together Dr. Viswanathan C. and Dr. S.S. Patel. I would also like to welcome the conveners of the session, Dr. A.K. Singh and Dr. Razveer Yadav. I would request Dr. Neelu Jain, Dr. Muralidhar Aski, and Ms. H.A. Bhargavi to act as reporters. May I now request Dr. A.K. Singh, Director IARI, to welcome the Chairman, Co-Chairman, and Speaker with the bouquet of love. May I now request Dr. A.K. Singh to introduce Chairman and Co-Chairman.
Very good afternoon to each one of you. It's my proud privilege to introduce the chair, and I don't have words enough to introduce him. You have already seen and listened to his wonderful presentation in the morning, and his lecture itself is more than the introduction that I can provide. So thank you, sir, for accepting to chair this particular session where we have 3 presentations. We have Professor Dhar with us, and Dr. Viswanathan and Dr. Patel. Dr. Patel and Dr. Viswanathan will be joining shortly. Also to co-chair this session, we have with us Professor B.D. Singh, former rector, Banaras Hindu University, and renowned geneticist, plant breeder, and expert in tissue culture. Professor Singh did his PhD from Saskatchewan, and he served at Banaras Hindu University as Professor of Genetics, Plant Breeding, and Professor of School of Biotechnology for long years. And his contribution to teaching, capacity building, human resource development in the field of genetics through his books in genetics and plant breeding is phenomenal. He is a household name. All the BSc Agriculture students, irrespective of whichever discipline they go, they have to go through the book of Dr. B.D. Singh. Dr. B.D. Singh has joined us online. Can you take him on the screen, sir? Can you have your mic on, sir, so that you can see once?
It's on. Ashok, mic and camera both are on.
Okay, thank you. Thank you so much, sir, and welcome you to co-chair this session. And to chair the session, we have Professor Balram with us. Now, I would like to request Professor Balram to kindly invite the first speaker with his introduction.
Good afternoon. Dr. Akhilesh Tyagi in the morning, there's an echo here, said that the program is dynamically evolving very quickly. In this particular case, not only do we have different speakers in the session, but we also have different chairpersons. I'm substituting for Dr. Mangala Rai, who is not able to be here. The very first speaker in this session is Professor Pawan Dhar from Jawaharlal Nehru University, and he will be talking on the dark genome. When I saw this title, I must say only one thing. For a long time, it was only the physicists who had these mega projects, and they talked about dark matter in the universe. Now, of course, the biologists have caught up with their genome projects, and there's large amounts of the genome that they don't know anything about, and that has now become the dark genome. And Professor Dhar, who also is in the area of synthetic biology, which is a cutting-edge field today, will be talking to us about the emergence of functional genomics 2.0.
Thank you very much for your kind introduction. And I'd like to thank the organizers Dr. Singh, and Dr. Vinod for having invited me here to share some of our work in this area. In fact, our work is slightly different than what you would expect. And I thought, to build some bridges between what we are doing in the dark genome space. I changed the presentation slightly, and I hope that it will gel with the theme of the event. So I thought we'll have some discussions on the, I'm trying to change the slides, yeah. So I thought in the beginning we can have some discussions on the Mendelian inheritance from a new perspective, and then move on and connect with the dark genome work that we are doing. So as far as Gregor Mendel is concerned, all of you know that he's very well known for these laws or ratios or principles or whatever people are comfortable in calling. But nevertheless, we have been using these findings for the last more than a century. The question that we are asking is that could there be a fourth law of inheritance that we are missing somewhere? If it is there, why did we stop at 3? Well, there is a reason for that. I'll come to that in a moment. Is there a use case for the fourth law of inheritance? If yes, how do we find it? And where do we find it? So if you look at the general classic approach, it doesn't apply to all the cases. So please keep the exclusion criteria away for a moment and stay with me for this thought. If you just look at the general pattern that somebody observed way back, and then let's say Newton, and then imagine something, and then experiments were performed. He didn't go to the lab to perform the experiments, but the logic was so sound and it worked so beautifully well that experiments were done later, but then he had by then come up with the equations and laws and so on. In biology, in general, again, there are all exceptions are there. In general, biology doesn't follow a top-down approach because there are a lot of contextual aspects in biology which are not there in physics. In other words, say for example, if a gene has to express, it is contextual. It is not constant all the time. It's fluctuating. So going back to this question of could we add something to these laws? The point is, what is the key idea here that Mendel has used? Of course, we know the pairs of contrasting characters and so on. But in my view, the central theme to his work was the search for biological constants. You see, it was like zooming out from factor to phenotype and zooming in from phenotype to factor. At both the levels, at one level, he assumed constant, that is factors. At the phenotypic level, he extracted the constants. And it took him several years to extract the constant. They're saying maybe 8 or 9 years, as per certain texts. I'm not sure about the number. Nevertheless, the whole idea is that his way of purifying the traits was nothing but getting hold of these constants that behave consistently as told. Because of which the 7 pairs of contrasting characters that he came up with, they were not influenced by the time. Now today it's raining, the color is changing. Today it's raining, the height is changing. It was consistent. So this was the constant, the power of constants. Because of which all he had to do is to do simple maths of addition and division to obtain laws of inheritance. Because of the power of constants. Now, if we have to find a new law, we need to hunt after constants. If you look at the Mendel's original paper, it's available on the PubMed, on the internet. The adjective constant appears 69 times in his paper. Probably it was his way of saying that constants are everything. Once you have a grip on the constants, now all you can do is play around with the numbers and we can get something. So the take-home message for us is look for constants if you have to find new laws in biology. Now, it's not necessary to go for laws straight away. One can start from organizing principles and then look for some regularities which stand the variation of the test across the labs, and then that will give us some idea about the constants. So now if you look at the constants, two levels of constant, one is the phenotypic level, the other one is the DNA sequence level. This is where he used the word factors. But between factors and the phenotype, information has to travel through an ocean of variables. Variables as in gene expression, protein interaction, protein expression, and so on, and also the cell-cell interactions. Now, the fact that information is able to travel this two major layers of variables and still consistently form a phenotype, it means that there must be some organizing principles at the layer of the variables. So let's hunt for those. So now if we look at what are the preliminary evidences in support of that, well, it's a grand challenge. It may or may not happen. We're not saying that it has to happen, but it's just an interesting search that that challenges you to search for some more regularities. So in this picture, I've tried to show that this is the work that we have done in the lab, that between two hub proteins, hubs are highly connected proteins in this case. So if you have highly connected proteins, how many linker proteins are in between? So when we plotted, this was done many years ago, by now the data has changed a lot. When you plotted the data of the hub connectivity, we found that the linkers are coming in a certain geometric pattern. There's a certain regularity. And this regularity is found across the evolution of the organisms that we consider, human, fly, worm, yeast, and pylori. We found that all of them are showing the same pattern. The interpretation from the graph is that most of the hubs have two linker proteins in between. And some of the hubs have got one linker protein in between. Is this a mechanism of robustness? That hubs don't talk to each other directly. They talk through their assistants, so to say. And this is found consistently. So could this be a template for something more interesting? Also, it has been proposed back in 2000, there was a Nature paper that talked about the power law in the networks. What you see in the far left top is the power law situation when you have log-log plot of connectivity versus frequency. If you have the lower right panel, that means there are more non-hubs in the network than hubs. And if you have the far right panel, this is a lot of hubs than non-hubs. And this is a random situation. So, but it was found that in biological networks generally follow this. Of course, there will be some deviations here and there. You cannot expect to get a straight line, absolutely everything falling on the straight line. That's the beauty of biology. So, between the constant layers of the phenotype and factors, you have two major levels of variables. And as far as I understand, systems biology was started as an approach to find organizing principles in the networks. Are there any languages that are consistent across the networks when proteins talk to each other or genes talk to each other through certain molecules? Is there any geometry? Is there any regularity? Is there any pattern? And if yes, can we catch it? People used, initially, I remember when the field was announced, back in 2000, there was the first conference of synthetic systems biology in Tokyo. There were more chemical engineers and mathematicians. And they were talking about how do we express biological data in the form of numbers? How do you connect life into numbers? So that's an art, but nevertheless, a lot of work has evolved in the systems biology space. There is a lot of lack of data for sure, Nevertheless, people have found interesting correlation. I'm not getting to that today. And the other part is, can we design these, some networks from the variable expression that we are getting? Can we design something starting from the part to devices to circuits and to multicellular consortia? Can we design something as an engineer? That was the origin of synthetic biology. This is a work in progress. It's not a finished story. I think it's going to take quite some time to find various flavors of these two fields. Nevertheless, people are working both in the process analysis, which is systems biology, and systems engineering, which is synthetic biology. I proposed it many years ago. Is this the time to construct a periodic table in biology? And many journals refused to publish, and there was a Journal of Biosciences from Bangalore that was very generous to accept it. Is this a time? Well, this is more of a thought paper, because you have the, if you identify the element of so-called bioperiodic table, then you got to have a data upstream and downstream, and a lot of data is missing. You know, you have a lot of gaps here and there. It is just a thought paper, so don't take it too seriously. It is just that, you know, if we have to find regularities, then we have to find the regularities across the spectrum at all the levels, if possible. Well, the main menu of today's talk was the dark genome. The unknown that we have still to grapple with, and in the variable space, I'm talking of an unknown in a different context. So, you know, traditionally, by converting genes into junk and garbage, we have learned biology, functional genomics. You downregulate something, or you throw it out and knock it out, and then you hope that it will do something in the cell and you will catch the function of the gene. There was a question that came up in our mind that there are three types of sequences, functionally speaking. One is coding for proteins. The other one is coding for RNA. Well, we used to call it junk DNA earlier, and Susumu Ohno, coined the term, and it became very popular, not because of the science, but because of the catchy term of this. And then people found that it was RNA coding. That's fine. But there is still some portion in the genome that encodes neither RNA nor protein. All right. So in our mind, this is the dark matter, though people are using the dark matter in genome or the dark genome in a very, very loose way, in a way that they say, well, if it is and not annotated, it is dark. No, we're not talking about those things. We are very clear in our definition. We are talking of those sequences that neither encode protein nor encode RNA. If you look at the human genome, or if you look at the yeast or E. coli, you have certain portions of the genome that are not known for either encoding RNA or protein. So the question that we asked was, can we do something from there? A subset of this question was also, why nature placed the signals at specific spots? How did she know that I have to place a start codon here and here? In between that, I have to express and this will do something. How did she know? Was this random, or was it intentional? If it was random, assuming it was a random thing and it worked, maybe many combinations worked. Some of the combinations didn't work over time, and we call them pseudogenes. They were retired in the course of evolution. But certain results were very much liked by the organisms, so they graduated as protein coding genes, and then they have RNA coding space. So if you have a situation like this, wherein this is a chance phenomenon, then theoretically you think of information at different levels, so you can theoretically make a lot of genes from here. If it was the one of the experiments that could have been done, it was done. Now there's a space for more variants. Is it possible or not? Can we make new genes from intergenic sequences? Can we make new genes from sequences? Well, I'll come to that. Those that were never known for any expression. So now to prove that we can make our own gene in the lab, our own protein in the lab, that nature probably did not make. When I say probably, we are matching with everything that's known from the NCBI database. But it does not mean that we know everything about what nature has created. Maybe these genes that we made in the lab, they must be made somewhere in some organism that has not been yet sequenced. So we are not talking about the entire construction of organisms. We are only talking about those that are identified, that have been sequenced, and we are with reference to what is known. Okay, so please keep that rider in mind. So what kind of sequence should we choose for making our own gene in the lab? We choose intergenic sequences from E. coli. Essentially what we did is the red colored lines that you see here, they are the sequences that were chosen in different orientation, overlapping, non-overlapping, different sizes. We did not know if it will work. So we took 2,500 sequences initially from the Intergenic Sequence database. We translated that computationally into protein sequence using the genetic code table of E. coli. And then we found whether these proteins that we're getting, the hypothetical computational proteins that we're getting, are they available from the NCBI non-redundant protein database? If they are available, even if there's a slight match, we discarded those sequences. So that was our filter. And then second filter was, are these sequences conserved in evolution that we are artificially converting into protein sequences? If they are conserved, we remove that also. Because today we might be tempted to make this, but tomorrow people might say that they have been conserved with a purpose. So are you sure you're making something brand new in the lab? So now what we did is, after that we got very small sample, and from there we randomly chose the sequences that you see on the screen in the form of red colored lines. And the numbers were all randomly chosen. We did not know it will work. So the odds against this idea were many. It was the first such attempt to our best knowledge. They did not resemble any known gene or protein, evolutionary non-conserved. And Jane Mark, one of the best tools in the town at that time, said there cannot be ORFs based on the certain data that is fed into the software system. So when I proposed this project to my students in Japan, I was working at Riken at that time. People were a little hesitant in accepting this new idea because they were already working on good projects. So they all refused to work in the lab on this project because they said there's no previous publication. So if I spend the next 2 years on the project and if I fail, who will be responsible? So we had to hire a technician from Singapore. She was a tissue engineering expert, and then Chao Su, she is the one who was hired as a technician, and she was trained in molecular biology. And for her, it's a salary, so she doesn't care about research. So she did all the work, and then she was the co-author on the paper. And a lot of people joined in. So the question is, how do you make a functional gene out of a non-expressing DNA? I'm not talking about RNA expressing. No RNA, no proteins. But it's a very simple problem because in the textbook, you know how typically a gene looks like. So if you install certain sequences upstream and certain sequence downstream, essentially you have made a textbook equivalent of a gene. But when you start doing this at the genome sequence level, it's a lot of work because you need to care for the order and the mutation every time you do some edit. We didn't have much time, and we were pitched against people who were publishing in top journals and so on. So we said we need to do something different. So we made the genes out of the genome. We did not make within the genome. So we considered this as an information. We thought that we will make a vector and we will install all these upstream and downstream signals on the vector. And then when you have to amplify the sequence and then insert in the vector, you're done. You don't have to care about the signals every time. You just care for once, and rest is just repetition. So when we did that, we started doing that. We were very happy to know that Invitrogen had released PBAT topo expression vector at that time. And that was a big gift to us because it had taken care of all the upstream signals. And in addition to that, the big gift was it has an his tag as an epitope. So which means that at the Western blot level, you have the antibodies, you can quickly see whether your protein is expressing or not, and it will be very specific to your protein. So we took the E. coli genome sequence, we did PCR amplification, vector insertion, protein expression, and really the eureka moment came when the Western blot showed that in presence of the inducer, gene was expressing and protein was getting expressed. And there was a very clear signal, plus and minus, inducer plus and inducer minus. This was a very big moment for us because till then it was all, in the air. Then the nature of questions changed. So the question was, do these polypeptides fold into a certain structure? Now, in our campus in Riken, Yokohama, we had 42 NMR machines. And every machine is totally booked for the Protein 3000 project. So it's 24/7. This was a big project in Japan, wherein Riken scientists had to solve the structures of 2,500 protein sequences, proteins, and rest of the Japan 500. It was a 5-year project, big grant, because of which we could not get access to the machine. So we said, let's stay with the computational prediction. And computational predictions, as you know, they come with their own limitations, but nevertheless, it was a little motivating for us to move forward and hope that there is some interesting story waiting to be unfolded. So the next question we asked was, what could be the phenotype of the cells? And if you catch the phenotype, you feel very good because it makes a good story for the journal. Otherwise, editors are going to say it's a boring experiment. So we found that out of the 10 sequences we took, we lost 4 sequences. We stayed with the 6 sequences. And out of the 6 sequences, one of the sequence, this is intergenic sequence-derived gene in E. coli, it was consistently killing the cells. So we were able to get one phenotype. We were very lucky. Now the question was, if this is true, and we did this experiment many times, so if this is true, can we restore the normal cell growth by switching off this user-defined gene? And that's exactly what we found, that as it starts killing the cells, 2 hours later, we switch off the gene and the cell growth is restored, as you can see here on the screen. So thereby showing a linear correlation between an expression and the phenotype. However, the story is not so simple. I tried to make it very simple, but then the vector that we have chosen, remember we didn't design it ourselves. So with the result that the insert was not exactly next to start codon. There was a big gap. The stop codon and the insert were not together. There was a big gap. That means vector itself was contributing its own sequence to the final protein sequence, which you can see here on the screen. The red colored alphabets, they are coming from upstream and downstream vector contribution, which means whatever you saw just now in terms of cell death, that is not the outcome of our experiment. It is the vector and the intergenic sequence collaborating to give this phenotype. So we have to do something to make sure that we have authentic results. So we changed the vector, we changed to PET expression vector. And then we said, let's add the promoter ribosome binding site and start straight away at the PCR level and add a couple of things here, with the result that earlier ECA3, why did we say ECA? Because Ekam in Sanskrit means first. So we thought that these are the first proteins, the first genes. And as you can see earlier, there was 50% of the protein was contributed by the vector. Only 50% was our choice. So when we changed the vector system, we found that it is coming to 0% vector contribution. Earlier, it was not showing any phenotype. And now we found that the cells are growing 3 times slower. So therefore, our approach then got scaled up. We felt comfortable with this new thought because now we have some evidence to play with. And at that time, the synthetic biology field was launched in MIT 2004, around the same time. And I was very fortunate to be there when the field was announced. And there were engineers from MIT who were launching a field for biologists. And I met one of the speakers, Ron Weiss, who was at Princeton at that time. And I told him, look, the field has just been launched and there's no journal. Can we start a new journal together? He agreed instantly. And we started Journal of Systems and Synthetic Biology with Springer. I ran it for 7 years, and then it ran out of steam. We had to close it down because by then other established journals caught up, and we were defeated. So, but nevertheless, this was a very good learning experience. And I can tell you it's not so easy to run the journal. I don't know how Professor Balram has been able to do that single-handedly for so many decades. It is really, really, really tough, but it is nevertheless very interesting and very, you feel that you're giving back to the community. So the functional study of the lab-made genes, as we call them, is the approach is overexpression. At the moment, you don't have a database of the knockout library database of non-expressed genome. When we talk of the database, we always talk of expressed gene sequences, not the non-expressed gene sequences. Now we are trying to show that even non-expressed gene sequences that we call as dark matter has got something, there's a purpose there. We're just trying to find the purpose. The ENCODE project has clearly established that a tiny part is proteome, bulk is transcriptome, and some of it is not coding. All we are saying is, could we convert the non-expressing sequences into functional genes and proteins? As a huge mega challenge. Well, to do that, we need some more proof because we cannot just rely on one experiment and think of a new area. So I'm expressing, I'm presenting some experimental evidence, 4 different experimental evidences that hopefully convinces you that the so-called junk DNA, the non-expressing DNA, can also be converted into products that we can use. And we considered Drosophila, E. coli, and Saccharomyces cerevisiae. So the first example I'm showing very quickly, these are short stories, is the intergenic sequence of Fly, the Drosophila melanogaster. And out of many thousands of sequences that we took, there was a huge computational pipeline. It's like a funnel, and you're looking for something and slowly decreasing the funnel. At the end of that, we got this peptide. And there was a striking structural similarity of this peptide, which is derived from the Drosophila intergenic sequence, with a commercial peptide, which is alpha defensin, which is an antimicrobial. And the structures are nearly same, but the sequences are so different. And when we experimentally made these peptides, we found that they kill E. coli, they kill Staphylococcus aureus, and Pseudomonas aeruginosa, thereby proving that the computational predictions do make sense. Second, we took intergenic sequences of yeast, again a large data, and then we ran it through the computational pipeline, several filters. I will not get into the details right now. We were looking for anti-malarial peptides, and so we tried to find something against these 3 targets. And so we took several sequences from the intergenic regions of yeast, computationally translated that into peptides and proteins, and then looked at the structural similarity with the known anti-malarial peptides. And when we did this is rigid docking study. This is not sophisticated MD simulation or even flexible docking. At the time, we had very little resource, and I moved to India. And we were just struggling to, get our basic facilities. So this is just a rigid docking. With that, we said that, okay, let's stay with these results and let's make them experimentally and see what happens. So when we found that experimentally close to 60% parasites were prevented from entering RBCs. Now, these are not designed peptides. These are not redesigned peptides. They have just been taken from the intergenic sequences as they exist. Then we asked again another weird question. Can we convert tRNA into peptide? Is it possible? Now, tRNA, as you know, has been doing a social outreach program for billions of years. taking the amino acid from one place and leaving it in another place and going back and forth and back and forth, never translated its own sequence. So we said, now this is not fair. Let's do something and see if tRNA can be converted to peptide. Now, in the cells, we cannot do that. We know there are certain steric hindrances and so many other reasons that will not allow us. But we again took this as a information, close to 100 tRNA species, different variants, computationally translated all the tRNA molecules, and then with a lot of filters, bioinformatics filters, we selected 2 tRNA molecules that were then graduated to the extent of getting chemical synthesis of their peptide sequence equivalent. So we converted the tRNA sequence into peptides, but again, after doing some computational pipeline. Well, this is the stuff that we did. I will not get into the details, but the most interesting aspect, when we got these tRNA encoded peptides, or T-reps as we call them, we exposed the cells to them, and a lot of cells, different kind of cells, and we got very striking results for leishmania. This was a big surprise because we found that close to 40 nanomolar, we are getting the IC50 value. There was a 66% inhibition at 40 nanomolar, and this was recently published in ACS Omega. This is just to show you that there is a lot of space that needs to be explored, and that's what we call as the dark matter. The other example I would like to present in front of you is working on Alzheimer. One of my PhD students was, he just completed his PhD on this topic. I'll very briefly talk about it. You all know how the plaque gets formed by the aggregation of these alpha beta derivatives from the original reaction when the beta secretase is basically, the whole idea is that in this figure, as you can figure out, the whole idea is that if you are able to inhibit base 1, you can probably prevent plaque formation. Okay, that's the whole idea. There are a lot of other details which I have not presented today. So again, it went through a lot of filtration. I will not come to that. And then we saw what could be the way to find those peptides. And finally, the experimental results is what I would like to draw your attention to. We have just completed the results, the experiments, and about to communicate the paper. We found 95% inhibition of beta secretase and the plaque formation. Now we are doing this in animals. Drosophila, one of my colleagues, is helping us in doing the in vivo experiments. But in vitro experiments were very striking, and we hope that we can bring some more interesting aspects of this work in the near future. So there are many other computational predictions that are undergoing experimental validation. We are trying to make functional proteins from pseudogenes. This paper was published, the computational work was published some time back, and this gave us idea of why not to bring them back to life. Likewise, we are trying to make some proteins, peptides from introns. These experiments are in the pipeline. We also got certain Cas-like protein hits coming from the intergenic sequence of E. coli. Reverse transcriptase, again, antimicrobial. The key lesson is that the dark genome is an untapped gold mine. This is our experience. We have some molecular inventory for the pharma and biotech. And the best part is that you own the IP of every molecule that you make. You do not make the molecule if it exists. You make it only if it doesn't exist. And once you have proven that it doesn't exist, you go and make it. So far, we have got all the successful results. I'm sure we're going to fail somewhere. We are waiting for that because that will be a new knowledge. We are trying our best to play with all kinds of sequences. I should not, maybe this is too early for me to say, but nevertheless, to bring some excitement into young minds, we are trying to make proteins from the antisense DNA. Okay, you have sense DNA, now we have antisense DNA. We have just started those works, and we also asked that can we make a reverse protein? So you have a certain orientation of the protein expression. Now, if you take it in reverse, would it make another protein of a different kind? A lot of computational evidence exists, which we have not yet published, which shows that part of these proteins have been reversed and it is in the PD. But can we make a full-length reverse coding protein, and what kind of function it will have? It's also for the fun and also for the fact that there will be some new scientific insight. So I was invited by European Science Commission back in 2010. 2009, the paper was published in a journal that nobody has heard, Journal of Biological Engineering. It was just launched with no impact factor, just indexed in the PubMed. Fortunately, they read the paper, and I was invited for the talk. And after that, I've been very fortunate to speak at many events, US National Academy of Science and all that. But the most interesting for me was the Organization for Prohibition of Chemical Weapons, OPCW. When I went there, I was wondering, it's like you're talking to diplomats from 80 different countries. And so one of the person asked me, I was wondering, why am I invited here? I have nothing to do with this event. So one of the person asked me, can you make a lot of these genes? I said, yes. Given funding, yes, why not? Then the real question came, can you make bioweapons? I said, well, this is something I didn't think of. But it was also scaring that, one can even go to this extent. So we've been very careful to make proteins only when we find some good use case and they're safe. When I say dark genome, I'm referring to essentially three kinds of sequences. One is a fully dark, that is the non-expressing. The other one is, we call it partially dark, the RNA expressing, because we want to take the long non-coding RNA to the protein level, that is the partially dark. And virtually dark, which is the pseudogenes, they were previously expressing. The question is, can we do this? And so I have tried to summarize this, just published this book, a small book on dark genome, a personal voyage of 15-year-old history into this. It's very affordable. So if you have this is the minimum, amount asked by Amazon. So I hope you have a chance to read it. We have worked on hundreds and hundreds of PhDs on protein coding genes. Imagine if we artificially express the 98.5% genome into functional peptides and proteins. This is what we call as functional genomics 2.0, artificially expressing sequences that were not expressing before, or artificially converting the RNA coding sequence into protein fraction. So can we build a knockout library of non-expressing DNA sequences? Also, can we artificially express the sequences? This, to me, is the unmet need of the community. We're also trying to find out in due course of time, are there any hidden linkage groups in the dark genome? If we're able to find, we have just started thinking along those lines, then maybe it will be new knowledge that needs to be uncovered. So everything started from, so to say, junk DNA, junk genes, RNA, and everything. So sometimes my students like to call this new area as junkomics. Well, after 15 years, when you have to hear this, it's very pleasant. These are the people who are involved in this work. I must thank Sakaki Sensei, who initially gave us the funding from RIKEN. And I must really heartily thank him, because if I would have probably submitted this project to DBT or DST or somewhere else, they always say, do you have a proof of the concept? Then we will give you some funding, which is fair enough, nothing wrong with that. But sometimes you need to get money even if you don't have proof, just for the sake of doing something out of the box. So he was very generous in giving the funding, and then other people joined in. So the unmet need right now is that dark genome has not been applied to plants. There is also going to be a dark transcriptome that has yet not been identified, and the dark proteome. As and when the funding comes, we are going to explore new and new layers, and we need collaborators and funding avenues. And If you are interested, and please join us. If you're not interested, please express your good wishes. We need a lot of them. Thank you very much for your kind attention.
Are there any questions? We have time. Yes, please.
Hello. Presentation was very interesting. I was just wondering about the new proteins, what you have translated and then shown some function. Have you studied its safety implications, particularly for humans or for any other systems, if you want to deploy them?
Which one are you saying? Can you repeat?
For example, one of the proteins, what you showed, has toxic effects on leishmania.
Well, okay, wonderful. I'm sorry, I didn't bring that story out. Leishmania story is even when we increase the concentration of the tRNA encoded proteins 10 times, human cells are not affected. Yes, thank you very much. Please, sir.
Sir, I have small two, three queries. One thing pains me extremely that the most important molecule like DNA in the all living beings has been described as junk DNA. Nature never produces junk, remember it. Nature never produces. Sure. And this is my request to Dr. Balramji, when he goes back, he brings all these scientists together. And because I got the solace when you use word gold mine, because in true sense, this is junk DNA. If Vladimir Putin allows us to live another 10 years, it will be the gold mine of information in so many ways. And it will precisely will tell you when the humanity started. Remember this. And I don't know how hesitantly you said about the periodic table, that don't take it seriously. As a scientist, you have to take it seriously till it is not proved otherwise. Mendeleev, who gave periodic table, he saw it in their dreams. And then dead of night, he got up, wrote down, and then you fill the gaps. This is how we do science. And you said that why nature gives regular gaps. Remember, nature behaves like human beings. It is extremely intelligent energy, which manifests with everything. And you have to understand this thing if you want to be a scientist. Otherwise, you will not be a complete scientist. You have brought out a lot of things interesting, and you use another word, DNA, the dark DNA. Sometimes we have a dark web, sometimes we have dark DNA. DNA is ultimate thing, we know it. What is your take on this? Thank you very much.
Thank you very much, sir. This is wonderful, motivating to hear this, very encouraging. We are just beginning to unfold a lot of insight that we get. The more we get into this, really, there is so much. I'm not able to answer your question in a convincing way, but I can tell you that there is so much I'm not presented today. It baffles me that it is there. It has not been uncovered. What is it doing there? It must have some purpose. So maybe we need to find new transcriptional mechanisms. Maybe these genes are used in the emergency. We do not know that really.
Artificial intelligence.
Right, sir. Yeah, certainly. We'll follow it up with that. Thank you.
If you are going to translate a transposable element encoding region, for example, then what would happen? Like, if the transposable element encoding region, if you do something on that, then it will create further multiplication or some kind of problem.
See, I am really not able to figure out at the moment intellectually what's going to be the outcome of that. All I'm saying is that we use a lot of computational stuff before we do even one experiment. See, first time we were very lucky to get that phenotype thanks to the contribution by the vector. If the vector was not contributing, probably we wouldn't have got anything. But now we are trying to process a lot of data and finally come up with maybe top 5 or top 2, top 3, because experiments are very expensive.
Because transposons and
Yes,
for
yes.
transposase enzymes. So
That's
in
right.
case if you do that,
Yes.
it may create more copies at the end.
So we are not we're not changing anything in the genome. That needs to be clearly expressed. We are not editing the genome. We are not changing anything. We're just pulling out the information and trying to see the purpose. That's all we're doing.
Pawan, just one comment on what you said. You mentioned antisense peptides. And I think you should look for work in the 1980s of a man called Blaylock, who worked on antisense peptides by translating the non-coding strand. And then he claimed at that time that the antisense peptide bound to the sense peptide. So he was looking for receptors. But eventually, I think that didn't hold up. I think we are more or less done
Sir,
for the first
I have
lecture.
a question.
A question? Sure, go
Only,
ahead.
sir, only one question.
One question.
I was wondering that have you the sequences which you have used, have you checked for the repeated sequences? Because when we say jump DNA, here are the plant breeders and molecular marker breeders are sitting here. So we more of interested in the repeated DNA. So the sequences which you have used, have you looked at whether they are repeated sequences or any random sequences are there which you have used for cloning and expression?
Which sequence are you talking
The
about?
sequences which you have used and you got the protein.
Yeah.
So I was asking that have you checked whether they are the repeated sequences or any random sequences? Because repeated sequences have their own importance in the genome. That's why I'm asking.
Yeah, if I'm getting the.
She's asking whether the nucleic acid sequence is repetitive, in which case you would also get a repetitive amino acid sequence.
Exactly.
What they call low complexity.
Right. So we are trying to see those sequences because as you go too repetitive, it's tough to the whole story. The kind of protein structures that you make, and this might become an exclusion criteria for the structure of the protein. We don't know that. But let's say ALU sequences, one of the sequences, and like that, some favorable material which is easy to do, wherein we have some hope of getting results, and after that we'll slowly increase the complexity. But really going for the easy ones first.
And second thing is when you are expressing, you must be having some promoter. So have you checked for the effect of the promoter which you are using when making taking the expression?
Wonderful.
Yes.
So the promoter is supplied by the vector.
Okay.
We don't use any new promoter. The same promoter goes for all the sequences. Of course, we can design the variants in due course of time. Also, whether we can convert the intergenic sequence into promoter sequences. Professor Manju Bansal's work, I was in touch with her from IC Bangalore. She has worked a lot in this area. So we were also wondering whether we could, design new promoters in future. Yeah. But yeah.
Thank you.
Thank you. Sorry, madam. One last. Okay. Yes, sir, please.
Good afternoon, sir. So, as in your presentation, you have talked about genome, dark genome, sir. So my question is, like, if you find your fourth law of inheritance in future by your research, then so what will be your assumptions? Like, what will you give the like the science in the science history? What will be your contributions by this the finding of the fourth law of inheritance? If in case you find the fourth law of inheritance by your research, then what will be your main goal by which you can create the history? Yes, sir.
See, I might retire by then, so that will be forgotten. But I guess there is already a regularity. There is an organizing principle because of which the factor information is graduating to the molecular expression and interaction level, and that is graduating to cell-cell interaction, and then you have the phenotype. So which means there is already a regularity. It cannot be random. You see, if it is random, then your gene knockout will not work all the time in the same way. That means it's random. But it's just a point in finding those regularities and then trying to build something on the top of that. Systems biology is a beautiful way of looking at the computational aspects of the network. A lot of work has been done. People have even created virtual cells. However, still that connection is missing between different layers. And I personally feel that even if you discover it, it's already there. You know what I'm saying? You're not designing something.
Actually, sir, by your talk, sir, I remember the quote of Elon Musk. Once he said, like, if we sequence a perfect DNA sequences, then we can achieve anything by the imagination, because imagination build the stand for anything. It's the main carrier for. So it's a nice creativity, sir, and you are doing a well job. So thank you so much for this kind of motivation and this kind of lecture, sir.
Thank
you
you.
so much.
Thank you very much for. Thank you very much.
Thank you for that. And I think we can break for tea.
Asha, you want to go? So welcome back after the tea session. Now we are going to start the two remaining presentation of this session, and I will request Professor B.D. Singh, former Professor Biotechnology and Genetics and Plant Breeding, and former Rector of Antisense University, to kindly conduct the proceedings. Professor B.D. Singh. We can't hear you, sir. Probably you are mute.
Oh, yes. Thank you, Ashok. Thank you.
Yeah, it's fine.
Thank you for giving me this opportunity to carry the session on. The second speaker in the session is Dr. Vishwanathan Chinusamy. He has done his BSc from Agriculture College and Research Institute, Tamil Nadu, in 1990. MSc in 1993 and PhD in 1996 from ICR-IRI New Delhi. He was Boycast Fellow during 2001 to 2002. Presently, he is principal scientist since 2010 and heads the division of plant physiology at ICR, Indian Agricultural Research Institute, New Delhi. He has been senior scientist since 1996 to 2010 and principal scientist from 2010 till date. He has several awards and honors to his credit. He has been recipient of ICR Jawaharlal Nehru Award 2001, TST Boycast Fellowship Award 2001, Fellow of Indian Society for Plant Physiology, ISPPJJ Chennai Gold Medal, Visiting Researcher, Department of Botany and Plant Sciences, University of California, between 2008 and 10, Visiting Scholar at the Department of Molecular Biology, Harvard Medical School, Harvard University, Boston, during 2009. His research areas are high throughput phenomics and genome editing. With this, I invite Dr. Vishwanathan Chinusamy to kindly come to the dais and deliver his lecture.
Thank you, respected Chairman, for your kind words of introduction. So, good evening to all of you. So today I'll be talking about genome editing. From the morning, I think several part of the presentation have been covered. So something may be repetitive. So please, I will just introduce what is genome editing briefly and some of progress made globally, and what is progress that has been made in India. So, as from the morning or from the yesterday we discussed, for any crop improvement program, the genetic variation is the basis, and the genetic variation can arise either because of the
And Dr. Vishwanathan, you are not audible.
Hello, am I audible, sir?
Now you are loud and clear now.
Okay, sir. Okay, thank you, sir. Good evening to all of you. Today I'll be talking about genome editing in crops. What is the progress that has been made? So as this is an important tool of crop improvement, and just before starting the topic per se, we need to use various different techniques to sustain the environment and sustain the human population on Earth. So we have to be cautious and judiciously use the natural resources. And as I presented in this slide, if you are not using natural resources judiciously, that may lead to like loss of human civilization, as these civilizations such as the Mesopotamian civilization, as well including the Indus Valley civilization. These have been wiped out because of the salinity and because of the drought stress. So the abiotic stresses are an important threats to the human survival if we use the natural resources unsustainably. And in addition to that, the other challenge is increasing population. So by 2050, we are expected to produce 50% more from the existing or slightly less land and resources. At the same time, we have to maintain the environmental sustainability. We have to reduce the greenhouse gas emission, either in agriculture or in other areas, the energy use. So to achieve sustainable developmental goals and to provide food and other requirement to all the population, we need to increase the genetic gain. And as a student of genetics, we might have very well aware that genetic gain, increasing the genetic, to increase the genetic gain, various tools can be used. And these tools are evolving every time. So we need to use appropriate tool to increase the genetic gain. So currently the rate of improvement in yield in the four major crops that has been shown in the graph is the maize, rice, wheat, and soybean. In this, it is currently the gain is only about 1% average, and we need to increase it to about 2.4%. For this, we need to use the efficient technology. On the other hand, in this graph, if you see, it shows the population increase and versus the resources such as oil or the phosphorus resources that are non-renewable, that are going to decrease. So we have to optimize the resource use. So towards this direction, scientists were from the beginning of the 19th century, systematic the crop breeding has started, and scientists were looking for various sources of variability to improve the crop. So this is one of the earlier we used the spontaneous mutation for improving the crop, and these are the two well-known examples. And just because of these two simple spontaneous mutations that brought green revolution, and it could sustain the population which has increased tremendously. So is there, since these mutations are very rare, and is there any opportunity to increase the mutation? And this slide morning, Professor Balram also presented, and also Professor P.K. Gupta, they have shown this slide. So scientists were looking for the mutagens that can increase the mutation frequency. So towards this, the induced mutations were discovered by Muller in Drosophila. He demonstrated that with X-ray, and Turner, he has shown it with maize. So the induced mutations were discovered, and it has been quite extensively used in the crop improvement program. Over 3,200 varieties have been released globally by using the induced mutation. And these mutations can be micro mutation or macro mutation. The micro mutations are listed above. That is, it can be small like deletion, insertion, or it is a substitution. Whereas in nature, there are the macro mutations, which include the deletion of large chunk of chromosome, or it is duplication, inversion, substitution, and translocation. So these are the various possibilities which you have well studied. So whether it is the induced or natural mutation, if you see the basis in case of the natural mutation, it is because of either some stress is there, which induces DNA damage. So during the repair process, these are not repaired properly, or it is by the mistake caused by the DNA polymerase during the replication. It can cause the spontaneous mutation. And again, various processes such as the crossing over and other chromosomal events, they can also by mistake lead to change in mutation. And the transposable element, as discussed in the morning, this can also cause mutation when it jumps from one place to another place. The induced mutation, again, they are either radiation or chemical mutagenic agents are used. All these induced mutagen, if you see the cartoon which is given in this site, it causes DNA damage globally. That is, in random places, it causes DNA damage, and this is the place where when it gets repaired, the mutation going to arise. And these there are several mechanisms are involved in the repair process, and this leads to when there is a mistake, the mutation arises. So the mutations are random and unpredictable, often rare with reference to the target gene of our interest. So precision mutagenesis was a dream of plant biologists. In all these cases, we are supposed to make DNA damage at specific locations. So scientists were looking for the tool for that purpose. When we use random mutation, this is an example from the project where Professor R.P. Sharma coordinated the project on EMS-induced mutagenesis. And when we use EMS, this shows how many number of mutations that can happen in the genome. So because these are not targeted in nature, so large number of mutations can arise. This is in each mutant. It was about 18,000 mutant, including the SNP and indel variation can happen. So large number of mutations occur randomly. And also when we do breeding, when we transfer, even by using molecular marker, when we transfer DNA from the region of interest from one genotype to other genotype, we often tend to transfer the extent of the gene that is required, the more than the what is required. So a lot of DNA is associated with the gene of interest is also getting transferred. Often we also exploit the translocation in our crop improvement program. This is the 1RS 1BL translocation is well known, and recently the other translocation, which is a region of gene from the Lamus racemosus, which is containing the biological nitrogen nitrification inhibitor coding region, is transferred from this wild cross into the cultivated species. So in all this process, the accuracy is less. So there were always the biologists were looking for the precise way of altering the genome. So this is an example by different method of crop improvement, how many mutations that can arise. This is in a study. If you are doing this selfing, in tomato, they have shown that 6 mutation can happen, and another study shows that there can be about 13 mutation possible. And if you do cross with wild species, depending upon the species, the number of changes can be very large. It can vary from 4 million or 7 million. And even between the genotype, if you make crosses, it is about 170,000 SNP variation can happen. And this is again in different study. You can see large number of spontaneous mutation can happen. In wheat, for example, the spontaneous mutation estimated number is about 238 per generation. So that's why scientists are looking for the precise way of editing the genome. So towards this, this was one of the earlier study where the effort were made to manipulate the genome or modify the genome in a targeted way. This technology was called as gene targeting, wherein precisely we can exchange a segment of DNA in the genome by using the homologous recombination. Although the work was started in plants and in animal cells, and the work in animal cell, it took faster shape, and that's why these three scientists they got Nobel Prize for medicine in 2007, and the work was started in about 1989. So at the same time, even from 1983, there were a lot of effort for using the homologous recombination in plant, but the success rate was very, very less. One of the reasons was that for any homologous recombination has to occur in normally in the cell. So when we transfer a piece of DNA into the cell, if there is a specific double-stranded DNA break in that specific place where we are intending to make homologous recombination, then it can occur very frequently. Otherwise, it is a rare event. So in that way, that is making precise DNA damage in a specific place was the major goal of this targeted mutagenesis. And this has been achieved by various approaches, and this targeted mutagenesis started from almost about 20 years back with the use of meganucleus. These are the endonucleases that are present in the cell. And these nucleases, they recognize certain sequences. So if you want to target this nucleus to any target locus, we need to modify the protein sequence so that it can bind to a specific sequence in the genome. So it is a very tough task, and it is very difficult to modify the meganucleus to target every gene. But there are successful products have been developed by using the meganucleus approach. And then this evolved further, wherein the zinc fingers are used. As the zinc fingers, they have specific binding sequence. You can alter the zinc finger sequence so that they can bind different DNA elements. And the zinc fingers are fused with nucleases, which are endonucleases again. So whenever the zinc finger goes and bind to the target region, the nucleus can make a double-stranded break or single-stranded break that can lead to the mutation. And another version of the zinc finger nucleus was the talons. Actually, the tail proteins are coded by bacteria, and these bacteria inject the transcription activator like proteins, and they have repeat variable residues, and these residues bind to a specific nucleotide. So we can engineer these tail repeats to target to any specific location. And then these tail effectors were fused to nuclease to reach the specific region in the genome to create the mutation. So these were the technologies, and they were effective. Only thing is the protein engineering and other things that were very difficult to follow for any normal lab. So then the discovery of the CRISPR-Cas genome editing has come in, and in the morning, Professor P.K. Gupta has explained this technology. So I need not again explain this technology, but this is an RNA-guided mutation approach where the RNA takes the nucleus to the specific location in the genome, and the nucleus cuts the DNA in the specific location. So Jennifer Doudna and Emmanuel Carpentier, they have been awarded Nobel Prize in Chemistry within 8 years of their original paper published in Science in 2012. So this technology is a very simple technology. We have two reagents in this. One is the endonuclease, the Cas9 enzyme, and another one is the guide RNA. So the guide RNA is a small molecule. It is only about 20 nucleotide in length, which is similar to that of the PCR primer that we use. But to make a binding between the Cas9 and the guide RNA, there is a scaffold RNA is that, which is about 40 to 90 nucleotide depending upon the nucleus that we use. So if you fuse the guide RNA to the Cas9, and which is called a single guide RNA, and these two reagents, if you supply into the cell, and the guide RNA can guide the Cas9 to a specific location on the genome, and then it can the Cas9 has the nuclease activity on 2 nuclease activity, which makes a straight double-stranded cut. So as explained by Professor Gupta in the morning, that the guide RNA need to be designed in such a way that it has a sequence which is called as protopesar adjacent motif. And the morning he was telling there is a limitation in this one because of the protospacer adjacent motif. So in case of Cas9, we need a PAM sequence of N and G, whereas there are several other nucleases are available which can use different protospacer adjacent motifs. So this limitation has been overcome. Now almost we can target most of the regions in the genome. So in case of Cas9, it just makes a double-stranded break, 3 nucleotide upstream to the PAM sequence, and sometimes we use double-stranded break. Sometimes we use single-stranded break also, as I will explain in the next slide. So the reagents can be delivered into the cell either by using the agrobacterium-mediated genetic transformation, or we can just use an RNA molecule of a single guide RNA and the Cas9 protein, which is a ribonucleic protein, which can be transferred into the protoplast, and where this can cause mutation. Then after that, we can recover the plant. So these reagents, when enters into the cell, they specifically target the specific region that we intended, and then it makes a double-stranded break. And this break can be repaired by different mechanisms. One is that if we make the break, after that we are not providing any reagent to the cell. So naturally the break gets repaired. So sometimes it will be repaired correctly, so there will be no mutation. Sometimes there is a possibility of error. So the non-homologous enjoining is an error-prone mechanism. That's why we get mutation in this one. So no repair template. It is an unguided repair, and these kind of mutation, maybe a few base pair insertion deletion can happen. So this is called as site-directed nuclease 1 mutation. The other kind of mutation which is possible is it is similar to that of the gene targeting. I told the first attempt to make precise mutation, wherein we use the repair template to guide the repair in the double strand cut site. This repair template is same as that of the gene sequence that is there in the genome, which we want to edit. Only we alter 1 or 2 nucleotide so that we can modify the protein, or we can modify the microRNA binding site, or whatever the intended modification is there. So this template which we provide, and by once the double-stranded break occurs, then again, because of the homologous recombination, the SDN2 type of mutation can happen. So this one will be exactly same except for the intended nucleotide change. In case of site-directed nuclease 3, the template DNA that we supply to the cell for repair process contain a foreign gene. So it is flanked by the homology region. So now the foreign gene can be integrated into the place where the double-stranded break is there. Then it can create the site-directed nuclease 3 type of mutation. In addition to that, as explained by Professor Gupta in the morning, the base editing system is there where we can induce C2T substitution, and in case of this adenosine deaminase, we can make A2G substitution. So in this case, if you see the figures here, we don't make double-stranded break. Here only single-stranded nick is made, and then the deamination occur, and then this one is subsequently it is by replication and base exchange repair mechanisms, the base is altered. So we can precisely alter the base. And this one also was explained in the morning how prime editing works. So here also we are introducing the precise mutation. Here, instead of providing the DNA template for repair purpose, we are providing an RNA template, and this RNA template is used to repair the cut site. So in addition to these two methods, there is another one method which is called as paste, which is drag and drop. This is another approach for inserting large insertion. That is, if you want to insert several kbs of DNA into the genome, you can use this approach, where the integrase is used along with the reverse transcriptase and Cas9 enzyme. So here it needs attachment site, which is normally present in the bacteria, and there are not many attachment sites that will be available in the location of your interest. So to create first the attachment site, we use the prime editing, that is the reverse transcriptase and RNA containing the attachment site sequence. And then we create the attachment site. after that, if you supply the DNA cargo, then the integrase can take the DNA cargo into the attachment site, and where large sequence of DNA can be integrated into the plant genome. So this is called as base genome. So in addition to this SDN1 and SDN2 and 3, which are the other mechanism that we discussed, that is the base excision repair mediated the base editing and the reverse transcriptase mediated genome editing. These two can also be put together under SDN1, because we are not providing any repair template to the cell. It is again unguided repair. So this can be come under the category of SDN1. This is important in terms of the regulation, as introduced DNA is there to serve as the template. So once then we come to the application of genome editing. It is limitless. Only like we need to have idea what possible modification that we can make, either for the product development or for to understand what each piece of DNA is doing in the genome. As discussed by Dr. Dar in the previous lecture, there are dark genome. So with the availability of these technologies, we can precisely remove those sequences from the genome in situ, and we can see what is the possible. So some of the possible modifications that have been done in the by using genome editing, this include the modification of the promoter. So often if we delete a particular gene, it may give phenotype under one condition, but if we do not know whether that gene is essential and whether for some other developmental function or for any stress specific function. So in those cases, the promoter elements can be edited. So one of the example is I will show one slide later. That is any pathogen, either maybe fungi or bacteria like Xanthomonas. They induce the sweet genes. They have a binding cis elements to which the bacterial transcription factor can bind, and they can induce the gene. So if you knock out those cis elements, then the bacterial or the fungal inducers, they cannot bind to the promoter region. So the gene expression, which may be otherwise essential for normal cellular function, will remain the same. Only under that condition it will not express. So that is possible. And sometimes if you want to change the expression of the gene only in the fruit, say if you want to reduce the expression of the ethylene in the fruit, so that we can make fruits that is delayed ripened fruit, that is possible because we know the cis element which are specific to fruit. So there are a number of opportunities are there. We can specifically either knock down the expression of the gene in a specific tissue or in the environmental condition or in the developmental stage. The other option available is in the 5' untranslated region, where the URFs are available, that is upstream ORFs. These are protein coding sequences. They code for a small protein. So presence of this URF limits the translation of the actual protein. So it tend to produce less amount of protein. So if you remove the URF portion, then the translatability of this messenger RNA increases. So we can increase the protein abundance. Then exon editing is most widely used. Either we can create a knockout, or we can alter the protein activity. That is, we can produce a protein that is resistant to any toxin such as herbicide, or we can enhance the activity. We can enhance the activity of protein by modifying its the inhibitor binding site or allosteric site. So many possibilities are there. Even we can engineer orthogonal receptors by using the exon editing. And sometimes splicing, alternate splicing is a very common biological phenomena. So we can edit the splice donor and acceptor sites, and we can see how the alternate splicing works. And also the mRNA, the microRNA binding sites is another novel targets that can be used to alter the expression. That is, we can modify the microRNA binding sites in the UTR, or if it binds to the exon region, that can be modified without altering the protein sequence, and thereby we can increase the transcript stability. So all these possibilities are available, and maybe more possibilities in addition to the editing of 5-methyl cytosine. That is, if you want to alter to study the DNA methylation, and if you modify this 5-methyl cytosine, then by using base editing, we can see what the role the epigenetics play in biology. So the applications, there are a large number of applications, whatever we think already it has been done. So this can be broadly like improvement of elite cultivar. It can be by correction of genetic defects, or improvement of the existing gene function. It can be for various purpose, resources efficiency, stress tolerance, and nutritional quality, or developing male sterility, or the seed production, haploid induction. All the traits have been edited. The other option is de novo domestication. So domestication took millions of years. So that can be achieved in a short period of time. And also you can develop naturally stress resistant plant. Like several land races, we know that they are highly stress tolerant. And if you want to improve the yield while keeping the stress tolerance intact, that is again possible by using gene editing, because we know several genes. In addition to the application oriented applications, this genome editing has huge application in understanding the function of DNA. So we can create any kind of alteration in the genome, and we can study how it works. So some example just I wanted to give. These are some domestication related trait which can be easily done. This is the Saturn1 gene, which can be edited by 2 different publications have been there. So we can edit and produce Saturn resistant rice. So these are the wild type rice which shatters its grain, whereas the edited lines we can see they are not non-shattering. So these are the natural mutations that can be created. The other one is the green revolution mutation that can be, again, it is created in different genotypes. This is the cultivated genotype, and even in the Varesa alta can be edited, and we can see this is a grass-like plant which can be converted into plant which can be looks like rice. So these are possible by using knocking out this particular single gene, the SD1, which codes for GS synthesis. And another one is the induced mutations that gave rise to herbicide tolerance, and this one is the Nagina22 Robin mutation that gives tolerance to imesothyper. And by using this mutation, our rice team, headed by Dr. A.K. Singh, already released variety. So this mutation was identified by using induced mutation in the project, which was earlier coordinated by Dr. R.P. Sharma, and now coordinated by Dr. A.K. Singh, by Robin. And so this mutation can be recreated by using genome editing approaches. Since we know that what kind of mutation that gives herbicide resistance, so that can be again recreated by using genome editing. So various options, like if you are predicting a protein function by using computational approach, even if we could not isolate a natural mutation, that again can be possible by using this approach. So some of the other application include the non-browning mushrooms, which was as early as in 2016. This was very famous. That of the 6 polyphenol oxidase, if we edit one of them, the mushroom browning can be reduced, and the mushrooms can be stored for the longer time. This is another example where the vaccimase is produced, which is almost 100% contain only the amylopectin, which is as huge industrial and other food applications. So this has been tested in the large locations, and it is about to be commercialized. The other application that I was talking is to reduce the expression specifically only under certain condition, not in all the conditions. So the earlier application, we completely knock out the gene expression so that it is always the gene is not expressing. So in this case, As an example, the sweet genes, they play very important role in development as well as resistant to diseases and various other activities it has. So if you are knocking out these sweet genes, the plant may not be productive. So in this case, as I told in my earlier slide, we can modify only the promoter region where the inducers that are coming from the bacteria that cannot bind to the promoter region. So there are 3 different sweet genes in rice, which has the effector binding element, that is Xanthomonas strain effectors, the tail effectors. They can go and bind to these sequences and they induce the expression of the sweet gene, which results in export of sugar, and that is used by the bacteria for its multiplication and pathogenesis. So if we specifically delete these sequences, then the plant can be resistant to this pathogen. At the same time, it is not affecting the normal function of sweet gene. So this is very well demonstrated here, and these are the wild type plant and compared with the edited plant, how they show the resistance in different cases. And again, another example in this direction was the ideal plant architecture. So in this case, they knew that where the tail effectors were binding, so they used a targeted approach to edit only that specific cis element. The other one gene which is well studied is the ideal plant architecture 1 gene, which is extensively used in the plant breeding program also. So this one ideal plant architecture, the enhanced expression of this gene gives the phenotype as compared to the earlier where the non-expression of the gene gives the phenotype. So we have to enhance the expression. For this purpose, what they did is they designed several large number of guide RNA to target the promoter region, expecting that some repressor binding site will be there. If it is deleted, then the expression of this gene will increase. So they could identify a 54 base pair deletion in the series of mutant that they have generated in the promoter region. It enhanced the expression and it gave the enhanced banical branching and the stem thickness, and finally the enhanced yield in the edited line. So it is possible to alter the gene expression only under a specific condition in the desired direction. It can be either we can increase or we can decrease depending upon the requirement. The accelerated domestication was well explained, demonstrated by editing of wild tomato to make it a cultivated tomato. So in this, 5 different genes were edited. This include synchronized fruit ripening, day length insensitivity, and the enhancing fruit size, as well as increasing the vitamin C level. So this Solanum pipinellifolium, it is naturally resistant to several biotic stresses, and it is also salt tolerance. So by using this wild type 2 different accessions, they edited and they could produce a tomato which produce a large fruit size and enhanced fruit yield. So instead of taking millions of years, they proposed that if we systematically do within 10 to 15 years, it could be possible to produce exactly the cultivated plant from the wild species. So this example again, some of the novel application wherein we can produce vitamin D. It is one of the major vitamin that is deficient in Indian population. So this also Professor Gupta morning explained. So this can be created, and we can see the amount of increase in the vitamin D that can be produced in this plant. This is provitamin, and this one can be produced vitamin D in the edited plant. So globally, large number of products are being developed. So this was in 2020, it was 231. Now it is crossed more than 300 products are there. And if you see the crops in which the edited products are available, rice is the one in the top, because rice is a well-studied plant, and the lot of effort that has been made in the genome sequencing and functional genomics and mapping of the genes, and all the effort it helped, because we know many genes in rice. So there are a large number of genes have been edited to produce large number of edited lines, and then it is followed by tomato, maize, and other crops. And in USA already about in last year already 168 applications have been granted like non-GM status under AMI regulated category. So in India also large number of studies are in progress. So this data I collected last year, the number of projects that are ongoing in India, it is about 30 different projects and worth about 30 crores. These projects are taken by various researchers. And this again, here also India also the scenario is same, and rice is the most worked crop followed by other crops. However, we need to increase number of people working in this one, and we need more resources need to be allocated. So this is the one project, the healthy crop project that I am showing. So this one project cost is 85,000 crores, whereas in India put together all the project they are only 30 crores. So a large number of people need to work in this area with more resources so that we can globally competitive in this one. This one in the April, once the guidelines were the approved, the Director General ICR called a meeting to know what are the crops and genes that are working. So in ICR, because of the historic decision of the government to exempt SDN1, SDN2, which are free from exogenous DNA, large number of scientists are interested, and several institutes are working. Hope soon we will get some good product. So in our lab, we are working on different genes, and today I will be presenting a small story on the DST1 where we have more data, whereas the other one we currently produce mutant and we need to analyze, and then we may be available later stage. So the drought and salt tolerance gene was of very much interest since it was published. It was published in 2009. So they used EMS-induced mutagenesis to identify the DST mutation, and this is after fine map they found that this mutation causes mutation in the DNA binding domain of the zinc finger transcription factor, which makes it non-functional, and this resulted in enhanced tolerance to the rise in the seedling stage. So the mechanism to a certain extent was also worked out. And later study, another study, it also showed that another mutation, it resulted in the enhanced panicle branching. And in this study, they have shown that DST is a positive regulator of cytokinin oxidase 2, which is known as the GN1A. So the loss of function of this one resulted in less expression of cytokinin oxidase, and thereby better grain yield under normal conditions. So this is a good candidate, because whenever we want to improve the stress improvement through various genes that are expressed under stress condition, often we find that the yield compensation is there. So we selected this gene, and when this paper published itself, I discussed with Dr. Mahapatra, our Director General, because that time he was working on screening the Nagina22 mutant. So we made an attempt to screen the N22 mutant population to see whether we can get a mutation which is in the DST gene. So we could not get a mutation, although we have got a mutation in some other mutation. We could not get a mutation in DST gene. So it is not always easy, even if we know that there is a possibility that we will get a mutation, there is no surety. So that's why we need to use the technologies which are more precise. So with the help of NASF funding in 2017, we started working on this DST gene, and we designed two different guide RNAs to edit the DST gene. One here and another one is here. The PAM site is underlined, and the triangle shows the place of double strand break. So we obtained the mutation, and these are various T0 plants, and we need to confirm by sequencing. Sometimes, since we used two guide RNAs, we can see if this particular region is deleted. We can also see the change in the amplicon length based on the terms we can identify. So now we identified about 5 different alleles of the gene. So whenever we are working on any regulatory protein, transcription factor or kinase or those kind of regulatory protein, it is expected that depending upon the place of mutation, you may get different phenotype, because they can interact with a different protein and it can alter protein-protein interaction. So that's why we selected different alleles of this DST gene, and this shows what is the change in protein that is made. And then we also analyzed phenotypically what happened. So earlier studies, it showed that it alters the stomatal sensitivity, thereby stomatal closure is there. And in our study, we found that these plants are more water is efficient. And then we examined, we found that the number of stomata are less in the DST gene in all the 3 different alleles that we examined, and the excess leaf water was less. And we also analyzed whether the altered stomata was due to the genes that are involved in the stomatal development. Out of these genes, we examined by real-time RT-PCR expression. We found that the expression of speech and screen, these 2 gene expression was less, and these 3 gene expression was less. Then we analyzed the promoter region, whether these genes have the DST transcription factor binding site. And we found that in the speech and the screen one, they have the DST binding element. Since this is a mutation where a large the C-terminal mutation was there, so we expect that this is non-functional, and thereby the reduced expression is there. So this plants, they showed enhanced tolerance to the osmotic stress and salt stress at seedling stage. This was earlier was also known. Then we examined under the adult plant stage whether it is giving tolerance to salt and drought stress. So this is the plants, adult plants, like in the how it looks after the minus 70 kilopascal stress when the soil moisture reaches. And we can see that this plant is more hydrated and it maintains better canopy temperature. And this is the final yield that we recover from these 3 alleles. So I told 5 different alleles. 3 of the alleles, 4 alleles were available earlier with us, and this year we added 1 allele. And one of the allele, it showed very different like behavior that we need to still confirm. That is, it produces higher yield under normal condition, but under the stress condition, its performance is it is very poor, or under the greenhouse condition, the seed set is very poor in that mutation. So in order to analyze the off-target effect, we identified 2 different mutants that are Cas9 free, and then we predicted the off-target. And here the first top line shows the guide RNA that is on the target gene, and the others are predicted off-target by bioinformatic analysis. And the red letter shows these are the letters that are not exactly same in the predicted the off-target predicted. So this was analyzed by DNA sequencing, and for all the predicted off-target, the sequence was same for different loci, whether it is wild type or the 2 different mutants that we have analyzed. So there was no off-target effect in this case, which means that the guide RNA was more specific and Cas9 was more specific. And the other requirement in the regulation is to show that there is no exogenous DNA in the edited lines. So this one, earlier the GABA tomato, which was already commercialized in Japan, they have used the nested PCR approach, where they designed the PCR primer for the entire vector that they have used. And by using this kind of PCR, they have shown that it is foreign DNA free, and this data is accepted in Japan, and this tomato is commercialized, and now it is also registered in USA. So we also designed various primers for the vector that we used for transformation, not only to the tDNA region, but also to the entire region that is covering the 15 kb vector is that we covered the entire region of the vector sequence. And we used the DST primer as a positive control. And this is the primary screening in the T2 plant. We identified some of the lines which are free of the Cas9 and the hygromycin, and these lines were selected and they are analyzed in detail. So this is the positive control, that is the DST gene itself, and this is the amplification from wild type, and these are the amplification from the mutant. So in the mutant, because of the 360 base pair deletion, the length is less, and then we used various control, like positive control is vector, and wild type was used as a negative control. And then the line from which it was segregated out, the parental line was used as another positive control, and the DNA free line that we identified was used as a test line. So in this, for various primers, we can see that the amplification within the tDNA region, when we use the primer, there is amplification in this line. whereas in the wild type and the DNA-free line that we identified don't amplify. And when we go outside the tDNA region, and we see that only the positive control, the vector amplifies, whereas other plants, they do not amplify as expected. So this was done for the other mutant also. Then also we examined whether these plants can be productive under the normal conditions. These are small plot trials that we conducted last year, and this year we may do a big plot trial. And this shows that DST edited mutant, that is different mutants, they produce more tillers and they produce more the panicle per hill. And they also have enhanced branching. And out of, as I told, out of these mutants, one mutant behaved differently. Still, it is not free from the transgene. So once it is free from the transgene, we will confirm whether it is how it is segregating along with the phenotype. So other three mutants, it is similar to that of the wild type. And they produce higher grain yield. So this, when we go to large field trial, we will exactly know how much the grain yield they can produce as compared to that of the wild type. So currently it looks very promising that it produces higher yield under normal condition. It produces higher yield under stress conditions. So we and also it uses less amount of water per unit amount of biomass. So the water use efficiency is higher. So from the mechanism point of view, based on the various gene expression, currently we know that because of the reduced expression of peroxidase 24 and the reduced expression of stomatal regulatory gene and the cytokine biosynthesis, the diverse phenotype that we found were observed. So currently we are doing a transcriptome approach to see the genome-wide changes in the expression. These are some of the work in the other laboratories in India that are doing in the, they are in the very final stage. This one is an example from the ICGP where the TLs, since they branched, one gene was mutated, and we can see the increase in the number of panicle and yield increase almost about 70%. Similarly, from the Indian Institute of Rice Research, Dr. Satyendra has edited the CKX1 gene of seed number. It is almost 2 to 3 fold higher in the edited line as compared to that of the wild type plant. In Nabi, Dr. Siddharth Diwari has used this approach to edit the lycopene epsilon cyclase to produce a banana which is containing more provitamin A. In DNA, Dr. Ravindran has edited the BADH2 gene to produce ASD16 with high aroma. So this is another example of reducing low glucosinolate line developed by genome editing at NIPGR. And similarly, there are work in ICGEP where the herbicide tolerant lines have been developed. So several labs are working. And the genome edited lines, they are non-GM as explained in the morning. Either if we use the genetic transformation approach, we can segregate out the transgene, and if you use non-DNA based method, there is no question of foreign gene integration. So in case of GM crops, the transgene is integrated and the transgene is there in the end product, and that gives the trait. Whereas in case of genome edited plant, the transgene is segregated out and this plant is discarded and only the mutant plant is carried forward. This mutant is the final product that we will be using, and there is no means to distinguish this from the conventional, like any other method that creates the mutation. So it is similar to that of any other plant that is developed by other breeding application. Because it is not containing foreign gene and the mutation is comparable to that of the mutation that are created either the natural or induced mutation. Several countries they exempted the SDN1 and SDN2 from the stringent GMO regulation. So these are the countries which are in green. They accept that the exogenous DNA free mutant as non-GM, and that can be used in the conventional way. Only currently the Europe is having the objection to this, but currently they are also under discussion. Now European Parliament is discussing this, whether we can sustain our agriculture without using this technology, because this is offer a lot of potential for resources efficiency and climate resilience. So they are thinking by 2023 they expect that the regulation may change in these countries. Already Switzerland and other countries, they are coming forward to use this genome editing approach. Our government has made the historic decision of exempting SDN1 and SDN2, which are free from exogenous introduced DNA, as normal product, and this is the pathway by which it will be regulated. That is, once we show the data that the SDN1 and SDN2 plants, they are free of exogenous DNA, then this will be considered as normal genotype, and it will undergo all the normal other varieties that are developed in the same test, and then finally the product can be developed. And if it is SDN3, then it will be considered as GM, and it will undergo all the regulation under the rules 1989. So these are some of the products that are already available in the market. This is high oleic acid soybean, which was developed by using a Talon approach. This is marketed from 2019 in USA. And this is the GABA tomato. This is also there in the market for more than 1 year. The gamma aminobutyric acid is enriched tomato is available in Japan. So and again, Japan took lead in commercializing the genome edited fishes. These two are two different fishes that have been edited in different genes for producing more biomass or more feed efficiency. They are also approved for the human consumption. So in summary, this offers a lot of potential to develop the genotypes at a faster rate. So this is not the only solution, but one of the important solutions that can be used to develop genotypes that are high yielding, are nutritionally superior, stress tolerant, and environment friendly. And it is also important functional genomic tool. In China, at least 2 different projects where they developed in rice, almost for 19,000 genes, they have developed mutation. So they know exactly what is the mutation and what is the genotype. So this will be useful for discovering new genes. In many of our crops, other than rice and few other crops, we don't know the function of the gene. So if we don't know, then we cannot use this technology more efficiently. So in case of oilseed and pulses, this one is very important one. And also we need a lot of human resources that work in this area. Whereas in other countries, if we see as shown in the other slide, large number of people are working, whereas in India we are very few people are working and very less efforts are being made. And other one is the patent rights, which is with UC Berkeley or MIT, and these rights are obtained by Parthiva. So these issue need to be solved once the product is ready, whether we can make it commercial. So it has the projected market value is about 36 billion by 2030, and it has a very high growth rate. So this can be one of the potential technology for improving the agriculture in a sustainable way. So I thank my students, Santosh Kumar, who carried out most of the work I presented on the DST gene, and other students who are working on different genes, Dr. Gopal Krishnan, who is associated with us in the DST project, and my co-PAs. And the funding agencies, the NASF, which funded the DST genome editing project, and the Department of Biotechnology and ERI for providing necessary funding and facility support. I thank Dr. Johann, who is my mentor, with whom I did postdoc, and he started genome editing in 2013. From that time, he was willing to start the work. So in 2017, I could start because of the fund availability. And mainly, I have to thank the Director General, because that time he motivated me to enter into this area and apply for this project. And I thank Dr. A.K. Singh for all his help and guidance. in this particular project, and Dr. D.K. Yadava for all his support through NASF project and otherwise also his support in this project. Finally, I thank the organizers of the symposium for giving me an opportunity. So when Dr. Singh told me that you have to make presentation, I was under tremendous tension. I see the name of the people, those who are making presentation in the symposium, and I was looking at how I am going to make presentations. So it was a great honor to me to make presentation in this August gathering, and so finally, I thank every one of you for your patient listening. If you have any questions, I'll be happy to answer. Thank you.
Compliments to you, Dr. Viswanathan, for a very nice presentation of the gene editing technology, the basics of it, the mechanics of it, the various applications, and the market products that have been.
Chairman, respected Chairman.
Please. Yes. Well, I was complimenting you for the very nice presentation.
Thank you, sir.
I think we have time for a couple of quick questions if the audience has it. Although what you described was lucid enough, clear enough, and detailed enough, but audience may shoot questions at you in the hope that better clarification will come. So can you handle from there the questions that will come, because I can't see if anyone is.
Yes, sir, I can see. I can see, sir. Yes, sir.
Please take the questions.
Yes, sir. Thank you, sir.
Dr. Viswanathan, you agree with me that in nature, everything is there with a reason, or everything happens with a reason. Am I right? So with this logic, DST gene is there in the rice plant with a specific purpose. So if we just simply, like, say, abrogate its expression, won't it have any other, say, beneficial or detrimental consequences because of that?
So it is possible, like we have to study in different conditions. So for that matter, all the loss of function mutant that has been there in the nature. So it is possible that in one condition it may recur. So we don't know exactly why that gene is there, whether it is recurred in certain condition or not. So many of the genes that we are using in the breeding, so it was there, but the knockout was there, but they are performing. So starting from all the green revolution genes, so they are there, but knockout still it is okay, and they are performing. So they may have functions. So currently we do not know. So only when we test under different environmental condition, then we will know exactly whether really they are important for productivity, or they are there for certain other functions.
This is principally because green revolution genes are structural genes, but here we have something which is a regulatory element.
Yeah, it is, see, structural genes, but they are regulating thousands of genes. SD1 mutation, if you see how many genes it regulates, it is more than 1,000 genes are regulated by the gibberellic acid pathway. So it depends, like only the time will tell what mutation is good and what mutation is not good.
Thank you.
Yeah, thank you. Nice.
Any questions that you can?
Yes, sir, one more question is there.
Dr. Viswanathan, when you talked about site-specific mutagenesis, I was wondering why can't we have 4 bases, so we have 4 specific mutagens. If they are available, wonderful. Why can't we synthesize and we knock down each and every base, and it is exactly far better and cheaper and easy than CRISPR, and can be alternative. But what do you have to say about it?
Yeah.
Because we have a long-term program with ex-Soviet Union, continuing with Russia, and ex-professor Balram Sharmaji is a boss. I think he's no more now. He was a great person in mutagenesis in the world. Rappaport, most of the people heard. Now he's no more. So we used to import before some mutagens from Russia, the department. And now also we can collaborate and we can synthesize it easily because the amount of technology available. What do you think about that, please?
Yeah, base editing is, it all depends upon what kind of mutation that we want to create. For certain purposes, base editing is very good. If you want to make a specific change in the amino acid sequence so that we can alter the protein function in desired way. And definitely it can be used as an alternative. But in some cases, when we want to make the loss of function mutation, or if you want to alter the promoter regions to modify the expression, then that may not be useful. So depending upon the application, definitely we can use it. And we are using SDN1 because it is easy to use it. Otherwise, base editing is actually the frequency of the mutation is very less. And so that's why not many publications in plants as compared to the simple approach where it is easy to use approach. Definitely those approaches are very useful. Technology is evolving. We will have more and more precision and technology improvement will be there.
I have a question on that question. The question is this, that the 4 specific base modifiers will act on bases present anywhere in the genome, not at a specific site. That is what I understood. Is that so?
Yes, sir.
If that be the case, then you will not be modifying a specific base at a specific site in the given gene. So it will be a random change in the genome.
Yes, sir. We need to target the base editor to the specific location, and it is often like the chances are relatively less as compared to the other approaches. So we can instead use prime editing, which is even easier than the base editing, where we can change the bases. Whatever we intend to change. So that option can also be explored in addition to the base editing. To change the bases, instead of base editing, we can use prime editing so that we can change the bases. But in both the cases, the frequency of mutation is very, very less as compared to that of the SDN1 mutation that we get by using the deletion approach.
Any more questions? If there are no questions, then I thank you, Dr. Vishwanathan Chunusamy, for very nice presentation, elaborate, clear, and explaining the basic technology, applications, product, regulation, and the advantages that one derives out of gene editing. This is a technology-driven breeding approach, but the new generation has to pick it up. And thank you for leading the way for carrying out the mutation work on rice, the DST gene. Thank you once again, and congratulations for the nice presentation.
Thank you so much. Thank you.
With this, I would like to invite the ultimate speaker of the session, Dr. Shri Kantias Patel, who did his MSc from US Harvard in 1997 with Dr. J.V. Gregor, PhD in genetics from IRI in 1980, diploma in principles of population improvement and hybrid development from CIMMYT, Mexico in 1995, and joined in teaching at US Harvard in 1983, where he was one of the celebrated teachers, I understand, and started working in cotton in US Harvard as cotton breeder. Served as principal scientist cotton in AIICIP system, served as head of cotton research station ARS Harvard Farm from 2015. He was professor and head of genetics and plant breeding, and later he became director of research of US Harvard. Currently, he is working as emeritus scientist, ICAR, doing research in cotton. He has tremendous research experience. He has been working in cotton breeding for 3 decades, developed over 15 cotton genotypes, registered with 3 color cotton types, namely medium brown, dark brown, and green, with PGRC, ICAR. He specializes, as you would see, from in cotton breeding, expertise in teaching quantitative genetics and population genetics, and currently teaching these courses in fourth year of BSc AG program. 40th year, he has been teaching for 40 years these courses. Awards received, he has been recipient of the best postgraduate teacher instituted by University of Agricultural Sciences, Dharwad, India. Professional Excellence Award instituted by Cotton Research and Development Association in 2020, and many others. He has served as RAC member of ICAR Central Institute of Cotton Research, Nagpur, and IIMR Hyderabad, published over 150 research papers in different national and international journals, guided over 24 MSc Agriculture and 9 PhD students. I had the opportunity to hear Dr. Patel in a conference in US Dharwad. I have read his papers and I have met him personally. An excellent teacher like you, sir, should need no introduction, but I took the liberty of introducing you. Now, please, through your lecture, you impress the audience and introduce yourself to them. Thank you very much.
Thank you, sir, for your nice words. I'm grateful to IRI for educating me. My guide is here, Dr. Sharma. And I'm grateful to Dr. A.K. Singh sir. ठीक निकाल ले देखता It's not consistent. Yes, as the title reads, I wish to speak in front of you about application of population genetics and, of course, quantitative genetics also in leveraging plant breeding approaches. Aptly, I have shown 4 representative crops representing random mating system, self-pollinated crops, asexually producing crops, and of course, often cross-pollinated crops. So, well, breeders have shown enough contributions, and we all appreciate the role of breeders in bringing the green revolution. And then in almost every crop, tremendous increase in productivity has been achieved, but still we keep saying there is stagnation of yield level, and the yield levels have to be doubled. A lot of bigger and higher targets are set for the breeders. And now, is it possible that these conventional breeding approaches can meet the bigger challenges that are there before us? How can you boost the confidence level of breeder? That is my special area of interest and specialization. So I'm always with those, if I had to categorically say, more than 90% of the breeders who work in different small towns with less than 2 lakh ORC to work with and hardly be able to have one labor and what kind of wonderful research we expect from them. How do we increase the efficiency of these common breeders spread across the country? Because they can only do conventional breeding, and how efficiently they can do this so that the genetic gains can be doubled. Ultimately, it means that. So so there are many speakers who have spoken about advanced techniques. We have so much to learn from them. But then we need to have some message for this common breeder who is left like an orphan, and then we have to care for them. How can we increase the efficiency of a common breeder? So now we do breeding. We think that we know quite a bit about plant breeding, but the problem is we still have enough complacency thinking we know everything about breeding. And of course, there's a lot of confusion, lack of clarity, and the picture before you is not clear. I mean, I've chosen him as a breeder, and then the vision is blurred and the breeding methods are not followed correctly. And the breeder is mostly unaware of it. And as a result, the gains are not achieved to the expected level. And unfortunately, these failures in studies are never reported, and no confessions of failures. So we try to develop new varieties, ultimately end up with lines we don't have big improvement, and we don't disclose, we don't confess before anybody, we don't try to understand why things went wrong. And there is no forum, no journal which accepts such negative results. If I say, I have done this and I have not got results, there is no journal text. First of all, you know, what kind of an importance is given to plant breeding system research? What kind of an acceptance journals normally show? All these are leading to very unfortunate situation. Let me not too boldly expose them. Now, with good knowledge of population genetics and quantitative genetics, which will provide a kind of spectacles to does it work? Yes, it takes a lot of time to change. So with the spectacle of population genetics, we expect plant breeders to have a very clear vision of plant breeding, very clear perception of plant breeding. The picture which was so blurred earlier now becomes very clear. And similarly to a breeder, a spectacle of population genetics brings much better vision of plant breeding. His perception improves and he will be in a position to bring out much better genetic improvement. And now I have tried to pick up some important topics of population genetics and quantitative genetics, and then a lot of derivations normally you come across when it comes to each topic. My focus is not on that. My focus is on what are the practical implications in plant breeding when we refer to each of these important topics. Now, I have just picked up selective topics only because when I cut down my extended summary, it with the great difficulty came down to 10 pages. So can you imagine if I had to have my full presentation, it may require 3 or 4 hours. And that's why I'm trying. It's a big challenge for me to cut it down to just 1 hour's presentation. I'll try my best. And I request you to cooperate. Yes. So every topic I have almost important ones. Now, how do you begin with? So we have broadly categorized these groups of crops, and we all know that the constitution of the population that is released as a variety is so different depending on the method of reproduction and mating system. It means the method of reproduction and mating system is so powerful that it determines what is the genetic destination a breeder aims at. The population ultimately he wants to develop and give it to the farmer. That is determined by the method of reproduction and mating system. How different they are, you have to appreciate that. A clone is what? A heterozygous and homogeneous population. Why is it like that in a sexually producing crop? Because the moment you find one potential plant, your search ends there. You create variability and identify single most potential transgressive segregant. your search ends there. You don't have to wait for stabilizing it genetically into a homozygous line because you're going to propagate it asexually. So the third part of third aspect of breeding, which involves genetic stabilization, is just not there when it comes to varietal development method of asexually producing crop. The story in self-pollen crops is so different. You have to create variability and you have to practice selection, identify the best single transgressive segregant. But your search does not end there. You have the third most important phase where the single most potential transgressive segregant has to be stabilized into a homozygous line. And that also is not a small job. So it requires several years' efforts. And that's why the varietal development methods of self-pollinated crops are so very different as compared to the methods of asexual producing crop. And then when it comes to, we don't speak of whether it is heterozygous, homogeneous. Is that the population that you develop must be an equilibrium population, so that the constitution remains unchanged when the crop, when the varietal population in the farmer's field is subjected to natural pollination. Hence, this third group of crops have a totally different target population to be released or to be developed by the breeder. So the three destinations are very different. Just as so for every genetic destination, the breeding methods are different. Just like saying if you have to go to Howrah, you have to go to Kolkata, you have to catch a Howrah Express. And if you have to go to Chennai, you have to catch a Chennai Express. If you sit in wrong train, you know where you end up with. Similarly, the varietal methods are dependent upon the genetic destination. In case of varietal development methods of asexual breeding crops, everything is so simple, just clonal selection or hybridization. And in self-pollinated crops, you are aware of hybridization followed by choices like pedigree, bulk, and SST. You don't have these methods enlisted under random mating crops. Why? Because the genetic destination is very different. So in case of random mating crops, your target is an equilibrium population, and that's why you follow population improvement schemes which enable you to come out with more potential equilibrium population. Hence, the journey is different, the methods are different. All the population improvement schemes that are enlisted in random mating crops don't find place in case of self-pollinated crops. That's why the mating system is so powerful. It not only determines the constitution of the end population, it also dictates what should be the methods used in reaching this genetic goal. So mating system also influences one more thing. Every minute step in every method is influenced by a good understanding of, a good knowledge of the genetic consequences of the mating system. That's why it's very important for us to understand consequences of different mating systems like random mating and particularly selfing. Now, is it possible that these groups, the methods enlisted under these groups of crops can be interchanged? Or is it possible that even some steps mentioned, these varietal development methods, can be adopted in improving other groups of crops? This is what is need of the hour. I would like to highlight these points here, because the methods, if we can adopt these principles in other groups of crops, some of these population improvement schemes, principles, if they get adopted in self-pollinated crops, we can have much better genetic range. And that's what I would like to highlight. Now, when it comes to consequence of random mating, we are all aware. We have a pool of male and female gametes produced by, let's say, a typical maize population, and there is random union of male and female gametes ensured by random mating. So when there is random union of male and female gametes, any male gamete can unite with any female gamete produced by the population. Without any bias and distinction, any male gamete would unite with any female gamete. And that's what makes the two events independent, and the product theory of probability works here. And as a result, square of the term, as you are seeing, square of the term containing allelic frequencies coming from female and male sides would determine genotypic frequencies of the equilibrium population. This is what is very commonly understood as Hardy-Weinberg-Castle's law. And then if we subject an equilibrium population to random mating, the constitution remains unchanged. And if we, that's why the target genotype is an equilibrium population. Now, if we subject an equilibrium population to random mating, constitution remains unchanged. If we practice selection and identify some very potential plants and use them for random mating, obviously we'll build a new equilibrium population, which is more potential than the existing one. And this is what is the essential genetic channel through which we bring about improvement and come out with new varieties in random mating crops. Now, the importance of random mating is well understood in framing these variety development methods. That's why you all appreciate that in every cycle of recurrent selection that is defined, you find one step of recurrent random mating, so that after every cycle you come out with a new equilibrium population, because the performance of the equilibrium population alone is reliable. You can trust the performance of an equilibrium population. During the course of varietal development, if we have generated a disequilibrium population, its performance cannot be reliable. For example, a hybrid itself is a disequilibrium population. You cannot pack hybrid seeds in a bag and label it as variety and give it to the farmer. It's a disequilibrium population. We all know that the farmer will get good yield when he grows this hybrid falsely labeled as variety. But the performance cannot continue because it reaches equilibrium and the performance of the F2 equilibrium population is obviously inferior to the variety hybrid's performance. So that's why before the population in front of us, when a builder looks at a population, he wants to trust the performance of the population. It's very essential that the population is at equilibrium. He must first know whether the population that he has in front of him, it may be productive, but if it is not an equilibrium population, he has to wait at least for one single generation of random mating so that it attains equilibrium and the performance becomes trustworthy. But we must appreciate that at least in recurrent selection schemes, that provision is made. There are still some varietal development methods in cross-pollinate crops where we don't subject the improved population to random mating. The simplest example is your mass selection, where we continuously go on practicing selection generation after generation, but we don't subject the selected plants to random mating and then wait for the population to attain equilibrium. So I will come to highlighting this small defect of mass selection and give a suggestion regarding how we should introduce a simple modification in the procedure. Yes. So I have already mentioned this. Any disequilibrium population is not trustworthy, and before trusting, we must ensure that the population is at equilibrium. Now, coming to the varietal development methods of self-pollinate crops, we have these steps of creating variability and exploiting it through selection, and continuous selfing is done to stabilize the most potential segregants into new transgressive segregant lines. Now, parents are recombined just once in a varietal development procedure. Hybridization, yes, we cross two parents. That's the only stage or time when we create variable by cross two parents and create variability. And we bank on only whatever variability is released by just one attempt of crossing. While in random mating crops, after practicing selection, getting good plants, again they are subjected to one step of recombination, which leads to an improved equilibrium population. This concept is very simple and straightforward, followed in random mating crops. Why is it not possible for us to introduce one step of random mating by using the best selection that you make even in self-pollinate crop? Because with one step of random mating, the population comes to an equilibrium, and that's not important. We create innumerable new genotypes. 3 to the power n genotypes are created in the equilibrium population, and many of which will be much, much better than
the segregants, the potential segregants we have used for random mating. And this is what is an essential new step to be introduced in varietal development methods of self-pollinated crops. We always tend to think, oh, random mating is something which has to happen only in random mating crops. No, this concept can be extended very much in self-pollinated crops. We have had many studies done in this regard. And then in my own thumb rules that I want to give later on, I would like to highlight on this point that you must have at least one additional step of recombination among the selected highest yielding plants while handling segregating generations in self-pollinated crops. That will make a big difference. The genetic gain is automatically enhanced through these simple modifications. Today in plant breeding, what is required is not doing something different, but the regular things to be done differently. And that's what my focus is on. Now, yes, that's what I have already highlighted. The best selections in early segregating generation, there is a very important necessity for us to plan this simple recombination. There are many crops where emasculation and manual crossing is not difficult. I can name crops like cotton. Even in crops like wheat and rice, it is worth trying this concept of random mating by using the best selection that we make in, let's say, F2 or F3. So without losing time, we create a new equilibrium population, which is much, much better than the potential of the highest yielding plant that we have picked up for random mating in early segregating generations. And I just picked up this 3x3 genotypic frequency matrix, while that we normally show while discussing about joint equilibrium and polygenic equilibrium. The point that I wanted to show is we may be using just 4 genotypes for in these populations, but when they are subjected to random mating, the first one that you see here, this is an equilibrium population. So these are the 4 different disequilibrium populations. The number of genotypes available are very few, but with one generation of random mating, we end up with an equilibrium population. And what's important, there are all possible 9 genotypes are created by us with one step of random mating. So the more potential genotypes thus arise. This is just an example of 2 loci considered simultaneously. Imagine you are picking up most potential segregants and you are considering how many loci? Hundreds of loci when you pick up best highest yielding plants. So imagine 3 to the power 100 different genotypes can be produced through one step of random mating involving the most potential transcriptive segregants observed in our field. And what is the benefit we get? Many genotypes which were not observed in these segregating generations will start appearing, and there are much more potential transcriptive segregants. Now, this is a very important topic which has direct bearing on hybrid development and many other aspects. That's why I lay importance to this. We have heard about the creation of allelic frequency differences between male and female pool of gametes. Whenever this happens, the population requires 2 generations to attain equilibrium. One generation goes in ensuring allelic phase equilibrium, another generation goes in attaining genotypic phase equilibrium. And there are some unique features of this G1 generation that is created when allelic frequency differences are created. What's unique about this G1 generation is that, first of all, allelic frequencies become same after one generation of random mating. Very interestingly, the population always falls above the equilibrium parabola. If this equilateral triangle has to represent populations, all the equilibrium populations that are falling on the equilibrium parabola, invariably, whenever we create allelic frequency differences and get the G1 population, or let me call it now as F1 population. Whenever we cross 2 varieties differing in allelic frequencies, obviously, we are creating allelic frequency differences. In other words, if you are developing a hybrid, what are we doing? We are creating allelic frequency differences and developing a disequilibrium G1 population, which we release to farmers as hybrid. So we have a new technical definition for a hybrid. What is it? It's a potential disequilibrium population, G1 population, developed by crossing carefully chosen genetically diverse parental populations where the genetic distance is wider, as wide as possible. Or in other words, the Y value is as wide as possible. The allelic frequency differences at each of these yield intrinsic loci are as high as possible, so that the frequency of heterozygotes in the F1 is maximized. So the concept of heterosis and hybrid breeding can very much be explained by using the idea of creation of allelic frequency differences and maximization of the Y value or maximization of the allelic frequency difference. What you notice is all these disequilibrium population F1s on just one generation of random mating will reach the equilibrium. And that's your typical F2. The F2 is at equilibrium and F1 is a disequilibrium population created by us. Why did we create it? Or in other words, let us answer a question. When and why should we develop a hybrid? Not in every situation we should create a hybrid. Under what circumstances we are supposed to use this technology? Because you are burdening the farmer. You have no right to burden the farmer unless it becomes absolutely necessary for you to do so. And when does it become absolutely necessary? There is high degree of dominance. Your heterozygote is much more potential than the homozygotes. Then what do you think of? You think of creating a population where the frequency of heterozygote is maximized. What kind of a population can you produce where frequency of heterozygote is maximized? Can you develop an equilibrium varietal population in cross-pollinated crops where frequency of heterozygote can be maximized? No. You all know very well that the highest possible frequency of heterozygote in an equilibrium population is 0.5. You just can't go beyond the value of 0.5 when it comes to frequency of heterozygotes and come to cell-pollinated crop. The value of the heterozygote is always higher than that of capital A or small a because there is overdominant gene action. So the value of capital A small a, when it's very high, you can't fix it in the form of a homozygous line. So both the crops or both the groups of crops, this possibility is ruled out. What possibility is ruled out? Exploiting overdominant gene action for varietal development. It is not possible. So when there is overdominance, very clear case of overdominance governing most of the loci influencing it, you are left with no other option but to develop a hybrid. In other words, you should develop a hybrid only when it becomes an absolute necessary. You can't cheat a farmer by giving a product like hybrid when it is possible for you to develop and by exploiting additive, partial, and complete dominant gene actions. Yes. Now, whenever this allelic frequency differences are created, it can be boon or it can be bane also. So how is it a boon? I have already explained. So we are creating allelic frequencies for betterment. In the sense we are developing a disequilibrium F1 population and giving it as a hybrid. This is where we're exploiting the phenomenon of creation of allelic frequency differences. So we're exploiting it for betterment. But yeah, here are some examples. Like what is the relationship between Y value and the frequency of heterozygotes? So the highest Y value that you see,
this is the What is not working? The red line that you see above, that's when the Y values. Yeah, that red line that you're seeing, what does it indicate us? When the mean frequency of the allele in the F1 is 0.5, that is when you get the scope, maximum scope for maximizing Y value. That's why when you cross two homozygous lines, the Y value can be maximized. Your one inbred is small a, the other is capital A. When you cross them, obviously, you get a 0, 1, 0 unique ideal population where the frequency of heterozygote is maximized. And that's what is the potential hybrid that you release to the farmer by crossing homozygous lines. Here, the Y value is maximized to 1, and that's why the frequency of heterozygote is also maximized to 1. So this can't happen in a varietal hybrid population. That's why we nowadays have completely switched over to using inbred-based hybrids instead of varietal hybrids that used to have in the past. So this I have already explained. 2 generations of random mating are required for the population to attain equilibrium. Hybrid breeding is just that. We get a disequilibrium G1 population and release it as a variety. So this Y is actually allelic frequency differences between the parents at a given locus. So it applies to all the loci influencing yield, and that's why this Y itself is called a genetic distance. This genetic distance Y value again figures when you come to the formula of heterosis in F1. You are all familiar. You might have derived this formula in your exams. HF1 is equal to summation of, let's say, dy square. And this Y represents the genetic distance, which is nothing but a allelic frequency difference between the parents at each locus, added over all the loci for which you are expecting over-dominance loci to be contributing to heterosis. So this Y again is figuring here as genetic distance. It means if you want to maximize heterosis in F1, if you want to improve performance hybrid, there are 2 pathways. One is maximizing the genetic distance, the Y value. The other pathway is to meddle with the D value and see if you can enhance D value. This is something unique. You may not even have visualized. Why enhancing Y is something straightforward. Enhancing genetic distance is what is actually done by breeders in the form of developing heterotic groups and exploiting them through different population schemes, population improvement schemes meant for hybrid, supporting hybrid breeding. So, as far as D is concerned, I'll talk about it a little later. Is it possible to enhance D? That's very challenging and interesting. But since the time is limited, I spare it at the end. If extended time is permitting me to touch upon this very fascinating part of how to increase D. That's again interesting. Let me see at the end. So how do we enhance Y? Obviously, as I said, we have to form heterotic groups. What are heterotic groups? These are groups of genotypes which are categorized based on high Y value between them, or already they are genetically diverse. So it's necessary to find ways and means of forming heterotic groups in different crops where you want to exploit heterosis. So this is possible. Many times, even I'll come to that slide where we have done it ourselves in cotton. We realize that when we cross robust genotypes with compacts, you get good heterosis in general. And you have, when high biomass genotypes are crossed with high harvest index genotypes, we generally get good heterosis. So the plant type groups themselves, if they are diverse, to some extent, they contribute to heterosis in general. So we can find out in your own crop what really works in this manner based on phenotype. Can you form heterotic groups initially? And then go for a fine-tuned formation of systematic heterotic groups. Once we form heterotic groups, it's possible to have heterotic boxes. What is meant by a heterotic box? The elite combiners of opposite groups can be identified, and you have a box of 2 parents coming from each side, in the sense A and B coming from one group and C and D coming from one group. These are elite combiners. And what do you mean by elite combiners? The predicted double cross performance or actual double cross performance involving AB from this group, CD from the other group will be higher. This is one simple yardstick for forming a heterotic group, not heterotic box. Because when I use the word box, it means you are involving elite combiners. And then once you form this group, it could be based on 2 parents from each group or even 4 parents. Then we generally call it as broad-based heterotic groups. So after forming heterotic boxes, we can exploit them by following population improvement schemes like, as I've shown here. Okay, first I thought, same thing I've already mentioned. We found that these are simple plant type diverse classes, which in between, when you make crosses, you get good crosses. And then between Shri green and high RGR, high relative growth genotypes and so on. Yeah, these are the population schemes we are all familiar with. Maybe recurrent selection for GCA, recurrent selection for SCA, reciprocal recurrent selection. These are already proposed in cross-pollinate crops or random mating crops, which support improvement of general combining ability. And in other words, they support for increasing genetic distance. The same concept, why can't we use it in self-pollinate crops? Obviously, we have done that. We applied the principle of reciprocal recurrent selection in self-pollinate crops like cotton by modifying the method to suit the mating system of the self-pollinate crops like cotton.
So, and just to highlight, because you're familiar with what is reciprocal reconstruction across pollinated crop, but then if it has to be modified and applied to a self-pollinated crop, these are simple steps we can follow. After crossing 1 and 2, coming from one group, you can go to maybe F3 or F4 to initiate your reciprocal selection for combining ability. Similarly, two parents from opposite groups also go to F4, and this is the stage when you can initiate reciprocal selection for combining ability. The opposite group's parents can act as testers, and initial screening can be done. And how do you mark completion of one cycle? As you see in random mating crops, the elite combiners of the opposite groups, between them can be crossed. So already these elite combiners have 4 lines that we have identified based on crossing them with testers which are opposite groups parents. So already it means you have tested the ability to combine with parents of opposite groups. So these elite groups, elite combiners from opposite groups, if you cross, definitely you are bound to get hybrids which are superior. So this principle has been applied and we have got very good results. One poster was displayed by one of my students in today's conference. Yes, so I talked about the boon and what is the bane. Knowingly or unknowingly, if you are creating early frequency differences, it will lead to a disequilibrium population. And this disequilibrium population's performance cannot be trusted. The simplest example is mass selection. We only know about mass selection that we select highest yielding plants, bulk the seed, and then grow, again select highest yielding plants, bulk the seed and grow, and continue this forever till we get satisfied, and then you give to the farmer. See, there is lack of clarity in the explanation. We're not realizing the fact that when we select highest yielding, let's say 20% plants, the problem here is the female gametes are contributed by the highest yielding plants. Agreed. But what about the male gametes? The male gametes are already contributed by the entire population because we are selecting for high yield after pollination has occurred, which means the entire population has contributed pollen. It's an inferior pool of pollen, while the female pool is a superior pool. Have we not created a frequency difference? Do you agree? Or you don't seem to agree? Do you agree? Good. So we are creating a allelic frequency difference with this simple procedure of mass selection. The tragedy is, whenever you are created, the population that we get next time is a disequilibrium population, and its performance is not trustworthy. But we still continue pressurizing it by practicing selection. And already it's a disequilibrium population. We again practice mass selection, select top 20%, bulk the seed, grow it again create a disequilibrium population. So we are continuously creating disequilibrium populations, observing them, finding them to be good, and feeling very happy. Oh, there is a genetic improvement. But have you confessed and confirmed that you must allow at least one step of random mating to see that it reaches equilibrium? Then you convince yourself, oh, it's good or bad. What happens whenever you create this disequilibrium population? Look at this. This was my base population. 20% plants, mass selection I've done. This is the population I've got. Oh, blown up like a balloon, looking very good. But the problem is it's a disequilibrium population. I don't subject it to random mating, and I hence don't know what is the performance of this good-looking population if I subject it to random mating and allow it to reach. It's not demanding anything from you. Just allow it to undergo natural pollination. This can very well be done even in an off-season without losing 1 year. You can very well use off-seasons when you're handling a maize program like this. You do mass selection, fine. The selected plant, just grow them in off-season and subject them to natural pollination. And that will lead to equilibrium, bring it back in normal season. Now the performance will be reliable. Otherwise, what happens is you're disappointed in this manner. You're disappointed in this. So look at this. This was a disequilibrium population. And if you allow it for random mating, the performance drops back. You'll never realize it. The books don't tell you that even at the end of mass selection, you should allow one generation of random mating to see that it reaches equilibrium and you are convinced that it is a good population. It's not mentioned. As a result, you end whenever you wish without subjecting it to random mating. You may even venture to give it to the farmer. So the performance will attain equilibrium after one generation of random mating. It will attain equilibrium mostly in the farmer's field, not in your own field. So that's a defect of creation of unknowingly creating early frequency difference. Why mass selection? In any approach of population improvement, if you are knowingly or unknowingly creating early frequency difference, if you find a population to be good, don't get excited and confirm whether it's at equilibrium or not. If not, use an off season, subject it to random mating, come back to see whether the equilibrium population is as potential as you found. So you are aware of the principles of selection response. They are very fascinating when it comes to random mating crops. I would like to ask you a simple question, whether if you select for high yield, will there be a response to selection? If you select for early maturity, because your direction of selection is negative, do you think the response is similar? So these questions we never address to ourselves. This is something interesting. And what really happens if the loci increasing yield are dominant? What happens if the loci decreasing what happens if the allele increasing yield is dominant, or what happens if the allele decreasing yield is dominant? Meaning, if there is positive dominance and if there is negative dominance, is there a difference in response? Okay, if you are selecting for a negative expression like early maturity, is the pattern of response different? So how do you relate gene action with the pressure of selection? The pressure of selection in terms of direction of selection and how much of pressure virtually you apply. So this is an interplay between these two forces, which ultimately determines, you all are familiar with delta Q. What is the delta Q you observe depending on the combination of this situation? This is something fascinating. And one important observation here is the response of gene action to selection differs in random mating crops as compared to self-tolerant crops. There is a straightforward relationship between degree of dominance and response to selection if you're talking about self-tolerant crops, if you're handling segregating generations and practicing selection. Higher the degree of dominance, higher is the deceptivity. And you're cheated, there is a mask of heterozygosity. I'm coming to this point a little later. But when it comes to random mating crops, the situation is slightly different. No dominance, partial and complete dominance, you get straightforward response. The problem arises only when there is overdominance. You see this subtle difference when it comes to response of gene actions to selection practiced in random mating crops and in self-tolerant crops. Well, this villain number one you're all familiar with. It's the environment. And then the observed phenotype is not reliable. I don't have to speak much about it. A breeder has to be very good in controlling the environment so that the environmental variance is reduced. So that's about villain number one. The villain number two is higher the dominance, extent of dominance that is there, which brings a gap between the genotypic value and the breeding value, or genotypic value and the value that's actually transmitted to the progeny. So we should be having good knowledge about this. And now coming to gene action versus response, because now I'm slowly focusing now exclusively on selection practiced in self-tolerant crops when you're handling segregating generations. How gene action influences your response to selection practiced in segregating generations, especially from F2 to F6. This is a critical stage, critical phase, because you have to have a very high proportion of heterozygotes in the population. And if there is high degree of dominance, it can be the heterozygotes look more attractive to you and you're tempted to select them. You can't talk to the plant, say, please tell me, are you a heterozygous genotype or a homozygous genotype? God knows when will you understand the language of plants. So till then, we have to bank on these methods of drawing inference about gene action and so on. So if the value is 20, it is reduced to 12.5 in the progeny. If the value is 10, what I mean by 10 here is if there is overdominance, there is a drastic reduction in the progeny performance. If there is complete dominance, yet there is a decrease in performance. Only when there is relative gene action, whatever plant you select, its genotypic value remains unchanged in the progeny. So that's the ideal gene action where the response is very good. And that's why you always recommend Pedigree method will be successful if most of the loci influencing yield show additive gene action and in the early segregating generations. And your selection will be a failure because if there is high degree of dominance, the most potential plants are heterozygous, they are deceptive, and you are tempted to select them, and the progeny mean comes down. And that's why you have different options of handling segregating generations. Pedigree on one side and single seed descent. You're all aware of why there is the method described as single seed descent. You want to avoid taking risk of practicing selection early segregating generations because we suspect that there could be some loci with high degree of dominance confusing you in the early segregating. To avoid taking chance, what is done is you postpone your selections to F6 to be on safer side. You start selection F6 generation. This is what is done in bulk as well as in single seed descent methods. But then can we still start selection in early segregating generations? So can we exploit overdominance for varietal development? I've already justified. It is not possible to exploit overdominant gene action for varietal development even in self-parent crops. Here, the simple reason that you see is the value of 20 that is shown by the heterozygote can just never be fixed. The highest value that can be fixed here in this example is only 10, and that is capital A. So the heterozygote superiority can never be fixed in the form of a homozygous line. You cannot exploit overdominant gene action for varietal development. Complete dominant gene action can also be exploited provided you're able to distinguish a heterozygote from the homozygote in the F2. That again is not possible, and that's why to be on safer side, you want to postpone your selection to later segregating generations. How long should you postpone? As it's described in SRT, should we start artificial selection in F6? How deceptive are the heterozygotes in F2? What is the extent of heterozygosity in F2? Does it reduce? At what rate it reduces when you move from F2 to F3? These are some interesting questions that should be, of course, arising in your minds, and always you want to find an answer for all these questions. So here are some answers for those questions. If the chairman permits, I hope I have not crossed the limit of time. You just have to show a red signal to me and I will stop it.
You may continue at least for 15 minutes, maybe.
Yes, sir. Okay. So, what are the safe zones for exploiting gene actions for varietal development? This is what I indicated. The safe zones for varietal development would be additive is the best. If it's complete dominance, well, if you can wait, safer side, wait till F6. That's why you have these methods. So should we go for pedigree? Should we go for single seed descent method? What is ideally required is to have a blend of these procedures. One step of pedigree, one step of SSD, depending on what is convenient to the breeder. So a situation-based modification, what is need of that's what is the need of the hour. So in the modification that I'm suggesting, I'm highlighting these aspects. So I have already indicated that this is the most potential transcriptive segregant when you're crossing these two homozygous genotypes. The most potential transcriptive segregant is now occurring with very low frequency in F2. And as against this, when you come to F6, obviously the chance automatically improves. And it surfaces out, and it's every observed genotypic value is reliable, and you can safely practice selection in F6. This is what is highlighted. And the risk of selection goes on increasing with higher degree, with the increasing degree of dominance. And then that's why you have suggested bulk. And then, yeah, how do you justify pedigree? See, more than 50% of the segregants are a junk. So why to carry them till F6? This is justification given in pedigree. And that's why you say, with all the risk involved in it, why don't we start F2 in an innovative manner so that even though there is heterozygosity, you overcome the problem. In this process, what I suggest is, so these are some comparisons of gene action versus exploitation or dominance, as it is shown there. I have already indicated it's very, yeah. So it's not possible to exploit complete confusion for the breeder when there is dominance, and he is disappointed by practicing selection early generations if there is dominance. while it's very safe and he gets good response and a continuous response if there's additive gene action. So the dilemma of a breeder is, as I said, should you start in F2 or F6? So the best is, as I'm going to show this, the modifications where you're blending steps of pedigree and SSD and bulk and so on. So now for that, it's necessary for us to understand what is really happening to the population after hybridization. What is the constitution of the population? What is the extent of variability at least in your F2 population? For that, it's necessary to begin with this best example. Normally, a breeder chooses two parents which are perfectly complementary, so that maximum variability is released for all those states for which the two parents are complementing. Can you get two parents which are perfectly complementing for, let's say, a dozen of characters which are influencing yield, quality, stress tolerance, and so on? It's very rare to get two parents which are perfectly complementary. So I must, for that give an example of perfectly complementary parents. I have to go back to the history and quote what happened after Chicago conference to Swamiji. So the most famous movie star of the Hollywood chased him virtually and begged him to get married to him. And her justification was that she's the most beautiful woman and he's the most brilliant person he has ever seen. And the most potential transgressive segregants can come in their family in the form of a child which is as beautiful as she is and as brilliant as Swamiji is. And that's the most potential positive transgressive segregant she was hinting at. But imagine, Swamiji has such wonderful genetic knowledge also that time. He warned the lady, your proposal is wonderful, lady, but what if you get a transgressive segregant, like an ugly boy like me with such a dull-headed, ugly boy like me? So your dull brain and my ugly looks, if they come together, that is also a possibility. So let's avoid getting married. So this is a range of transgressive segregants you get in the F2 itself when you cross two parents. When they are perfectly complementary, you get the entire range of transgressive segregants. They could be positive, they could also be negative. We are aiming at picking up the most potential positive transgressive segregants. And that's what is essentially done in a breeding effort of varietal development. In this process, look at the simplest example. If the two parents are differing for only 21 loci, can it happen? It can never happen because you have got dozens of characters influencing yield, quality, stress, and so on. The number of loci n would be running to hundreds. So if you have created variability, let's accept it is n equal to 21. 3 to the power 21 different genotypes are created. And to observe all this entire range of genotypes, the minimum population size required is 4 to the power n. And what is 4 to the power 21? I remind you again, 21 is a silly number. Actually, it should run to, in realistic situation, hundreds of loci. Let's presume it is 21. So 4 to the power 21 is 4 trillion. So the minimum population size that you must raise in F2 is 4 trillion. This is not optimum. Optimum has to few times more than this. Okay. If the number of loci for which the F1 is segregating runs to realistic number of hundreds, the population size that you must raise runs to trillions and trillions. The entire continental area will fall insufficient to raise one F2 population of your class. Leave aside your own institute's land. So let's first accept the optimum population or even the minimum population size we are not able to raise. If you ask the breeders, I have heard them saying, somebody when asked, what is the population size you normally raise? My Punjabi friend said 50. He was very confident. And I had a Chennai friend. He said, simply 200, sir, big number I am raising. See, we are not confident about what is the minimum population size to be raised. Without having any knowledge, we are raising 100, 150 plants and be very happy about this. And what is the impact of raising a very small population size while the minimum populations are required is very huge. The sampling effect is terrific. And what it does, you'll see it in the next slide. Actually, a breeder churns the genetic backgrounds of parents with the hope that he gets a transcriptive segregant equated to the butter that you get when you churn the curds. So you create the transcriptive segregants by proper churning of the genetic backgrounds of parents. So I have told you the minimum population size is just hundreds, while it is running to trillions. Let us accept that we are raising a very small population size. Because of the sampling effect, we never get the most potential transcriptive segregants in our population. It becomes a story of this person who is heading towards the sea with a small bucket. And everyone is surprised to see somebody going towards the sea with a bucket. And he says, I am going to collect the sample of seawater and study the entire range of fauna existing in the sea. Then when he asks, what is your size, sample size, how are you going to collect? In my bucket. So he imagines to collect the fauna ranging from jellyfish to blue whale in a small bucket of water. So we laugh at him, yes or no? But we don't laugh at ourselves. We are raising exceedingly small F2 population, hoping to get the most potential transcriptive segregant to be seen in that small bucket of 200 plants. This is the tragedy with us. And we don't realize we are committing blunders. So now, accepting that you can't find the most potential transcriptive segregant in the F2 with a small population size, what can be done? Let us accept it's not there. In that small size, the best segregant is not there, simply not there. What best can we do? So the situation is comparable to this. Every time, because of ignorance, the heavy luggage of ignorance, you go to the railway station, and every time you go, by the time you go to station, the train is gone. And what is the train? The train is of transcriptive segregants, which has left the station much before you went to catch it. This is exactly what happens when we raise small F2 populations. You always miss the most potential transcription. Let us accept you are missing it. What can be done? How do you catch the train which has left the station? How do you recreate the most potential transcriptive segregants that are missing in the small population size? See, there are different ways you miss the train. I mean, the most potential transcriptive segregants. So what can be done to recreate? The best thing to do is the best segregants that you observe in your F2 population or F3 population fall in this category of 10 to 15%. They have bits and pieces of the missing most potential transcriptive segregant. Bits and pieces of the genetic background they have. They may not have the complete genetic background of the ideal missing most potential transcriptive segregant. To make it convincing, we have to give examples like this. Then only students get interested in this. Here is a fanatic fan of this movie star. Most of us are also fans of her. And then if the poster is torn to pieces, what an ardent fan would do? Desperately, he will join these pieces and try to recreate the poster. It all depends on how crazy he is, how fanatic he is about the ideal, the idol star. And we as breeders should also have that craze to recreate the most potential transcriptive segregant which is missing in your small F2 population size. It is possible. When is it possible? The available best segregants, just as you see an eye, the nose, the lips, and the ear in some of these, similarly talking in terms of bits of the genetic background, yes, they are there. Bits and pieces of the ideal genetic background that you wish to recreate, they are existing in your higher staging plants of that small population. So what's necessary? You must now subject them to one step of recombination. You should recombine, or let's call it as random mating, involving the most potential transcriptive segregants that are visible in the small population, so that you can recreate what is missing. This should be a mandatory step in handling segregating generations. So this can be done in F2 based on selections, or in F3, or even F4, any of these early segregating generations. So we have done such a study, and I don't have to explain. The results are very encouraging. So it means we must have one mandatory step of such recombination to get the most potential transcriptive segregants which are mixing in our small population size. This is what is a routine step in random mating crops, but we must introduce it in self-pollinated crops. So coming to the myth about homozygous genotypes, everyone says by F6 we can start artificial selection because plants are homozygous. I think most of us have that feeling. So let us ask one question. If your F1 is segregating for realistic number, let's say something like n equal to 300, that should be the normal genetic background for which your F1 is segregating. And what is the question asked? What is the proportion of homozygous plants in F6 generation when you decide to start artificial selection as dictated in SST? And what is the answer to this question? Many of us may quote a figure like 90%, 80%, 70% proportion of homozygous plants in F6 when n is realistic, like 300. You'll be shocked to know that the proportion of homozygous plants in F6 when n is realistic number like 300 is this. An unforgettable figure, 007, of course, a point before that. 0.007% is the proportion of homozygous plants that you see in F6. It's such a shock given to, a rude shock given to the breeder. He tends to think that all the plants in F6 are homozygous. No. Should we wait till F6 to start artificial selection? How is it possible? If you have to wait till F6 and start artificial selection, when can you release more varieties and become a vice chancellor or a director? It's simply impossible. So what is the solution? We have a very nice convincing genetic principle or explanation here. If not homozygous plants, by F6, plants are nearly homozygous. So we must, as breeders, understand the concept of nearly homozygous plants. What it really means. If n is 300, we should accept plants which are homozygous for almost 290, 91, 92, up to 95, 96. If there is residual heterozygosity for about 2, 3, 4, 5, 6, up to 10 loci, we should not mind. So this is the class of genotypes called as nearly homozygous. Yes, their proportion is more than 90. And you can accept, by accepting nearly homozygous plants, start our artificial selection in F6 generation. If you are doing it, we should also accept that there is residual heterozygosity. Many times we stop within line selection by F4 itself. We start bulking the lines by F4. Some people bulk in F3 itself, which is a horrible step. So we must remember, even in F5, F6, if possible, try to have individual plant selection picked up from these potential lines. Never bulk lines in early generations, not even in F6. If you go on bulking, presume that already it is homogeneous. We see the end results. Many of our released varieties are not genetically uniform. You get a lot of residual heterozygosity, whether you are selfing and deriving a variety or you are backcrossing and deriving a new line. So if you don't take care of these aspects of residual heterozygosity or residual background of the donor parent, you end up with problems. The end product is not what you expected. So that's about this. And then
could we move towards the end?
Yes, sir. End is quite far away, sir. Absolutely no problem. My only objective was to speak as much as possible and just show a trailer about the whole aspects that I have. If you have observed, the extended summary was 10 pages. I could not bring it to a still shorter version because there are so many interesting topics. I really don't mind because I just think it is part 1 of my lecture. So I'll be waiting for delivering part 2, part 3, part 4, and so on in the coming days. And I have a good friend and well-wisher in the form of Dr. E.K. Singh, sir, who trusts me, I suppose, and we'll have many sessions in the future like this. Thank you. Thank you. Thank you very much.
Professor Patel said, I hesitate to say words of praise simply because no words will be enough to praise you. The lecture that you gave us the basics of mode of reproduction, pollination, gene action, population handling, breeding scheme, and all their relevance to the possibility of getting the end product, that is an improved version of either a genotype, pure line, or a heterozygous population. Listening to you was a real treat, and I think everyone must have enjoyed it. If the audience wishes to interact with Professor Patel, I think he will be very happy to take questions. Sir, you will have to handle the questions from
Yes.
the audience.
Yes.
Sir, just recently a new approach called MAGIC or multi-parent generation cross-site transformations are being touted as one of the ways to increase the recombination and work towards population improvement. What are your views on that?
Yes, yes. That's what I could not show those slides. That's what I'm saying. The thumb rules I was to give at the end, that you will never find two parents like Swamiji and the Hollywood star when you have to create variability. Normally, the traits are about 10, 20 for which you want to create variability. And you can't find two parents perfectly complementing. You have to pick up a team of parents, like a cricket team or a football team. So you will have a bowler, you will have a batsman, you will have a keeper. Exactly in that manner, you will have a team of parents. Some of them will be good for abiotic tolerance, some of them would be good for quality, some good for grain number, some good for grain, grain quality, grain weight, and so on. So the choice, the idea, this is something important. I don't mind spending 1 minute on this. They were all existing in the slides. So a team of experts involving breeders and entomologists and pathologists, all those who matter, should sit together and see the scorecard of the leading genotype, identify the team of parents, and then involve them. In fact, you should not leave to that simple orphan, a small breeder in a small town, to have his own germplasm, create his own variable population using 10 parents, 20 parents. How can you expect? It should be a centralized effort, a coordinated effort at the national level to create variability like this by involving a team of parents. And this population must be distributed among breeders. Then if you have 10 breeders, 10 into, let's say 2,000 will be 20,000. At least the total population raised will be 20,000 now. In this process, somewhere in some center, you will get good segregates. And they must be subjected to one more step of recombination. These are some innovative modifications. A lot of them have been suggested. I think luckily you have that extended summary which covers all these. Otherwise,
Yes, Mr. Speaker, we have learned an interesting lecture. You appear to be a very, very practical plant breeder. But one thing I would like to say that self versus cross-pollinated crops, say random mating in self-pollinated crops, you can imagine how difficult it is.
That's why I'm saying,
how
sir.
difficult it is.
It is not difficult, sir.
It will take a long time, double the up. Now, in both the crops, both set of crops, you have the selection of parents. The story starts from the very beginning. You know how efficiently you are selecting the parents. There are different methods. There are quantitative methods to select the parents, the parental selection, diversity, the geographic and genetic and all kind of diversity. That starts from the parents. Then selecting the transgressive aggregate. I agree with your small population, which is a general defect in our general selection.
Defect starts with choice of parents. I think you also have got the point.
The choice of parents is the most important one, but then we have to reject, we have to go away, made a large number of processes, very large number of processes. And then among the large number of processes, we have to use, first we have to get a visual wisdom, besides your parental background, and then you have to reject a very large number of processes that some at F1 level, which are not showing any heterosis. See, there is heterosis maybe that will contain, the identity or expressed into the baby factor, fixable factor. So that we cannot distinguish at the F1 level. Then when you grow the F1, you will immediately know which of the processes are gaining and which are likely to give us from this side, isn't it? So those are the classes which are the need of the breeder. So those classes should be very, very logically and very, very intensively studied, and the F2 population we can just make out whether F1 versus F2, what is the performance of those classes. The F1 and F2 performance of the selected segregate is concordant. You are doing that there's going to give you from this segregate. So that's how you can pick up from this center. Yeah.
One point, I don't know if we are differing with respect to possibility of random mating. There are many crops where manual emasculation and crossing is not difficult, but it is a very mandatory step to have random mating involving the highest yielding plants that you find in your segregating conditions. This we don't think of, but it is required. And I would say, I go one step ahead and say, where is plant breeding system research happening? These are the objectives you must put in having simple experiments given to students. What if you have highest yielding plants selected and you subject them to random mating? What is the performance of the population that you get as compared to when you select these best plants and then test the performance of these selections? This kind of simple research is required, badly required in plant breeding.
I think there is no disagreement on that.
No disagreement, but
it's pretty much agreeing. All that you are saying is that when we are growing the F2s, along with that, we also have the F1s, and only the performing F1s, and we should focus on those F1s for advancing the generations. I have one small, submission, and that's with regard to the population size. While I'm not disagreeing with you, and I also teach the way you have said in my class, but one thing that intrigues me, and probably the chair might, clarify, or you can clarify, is that if you look at most of the molecular mapping program today, irrespective of the crop, you start a cross between two inbred parents, and you follow single seed descent method. Take about 500, F2s and advance them to a recombinant inbred line population, fix them. 500 is a fairly good number, and there are umpteen number of reports where 250 to 500 RILS populations have been used, and they are good enough to capture the major QTLs which are segregating in that population. So you hardly get about, say, we have done, many of my students have worked in rice, where we have got as many as 50 to 100 QTLs captured for different yield and yield components in a population size of 310. Now, if that is possible, how is that possible? And also in most of the speed breeding programs, which are being emphasized using the facilities like speed breeding facilities, the population size per cross that is being, forwarded following rapid generation advanced approach is close to 500. So nobody is looking to the kind of calculation which is presented also in Jones calculations. Yeah. So that is something not possible. So something which is not possible. Definitely. I think we must work out what is most practical and most doable with a result.
Yeah.
Thank you.
What I said was, we all know that we can't raise trillions of plants. We are raising smaller population, but let us try to have as large as possible. Let us not stick to 50 or 200 plants in F2. Let's try to have at least few thousands of plants. The best way is I was to show in the next slides, since the minimum population size in F2 is higher and it reduces considerably if you go to F3. This is the biggest boon you get. If you avoid selection in F2, the big advantage is the extent of heterozygosity is drastically reduced in F3. This is one big advantage. And next is the minimum population reduces considerably in F3, which means the chance of getting the most potential transcriptional segregant increases considerably in F3. So today we have to think, why should I select in F2? Why don't I dump my F2 in off season without wasting time? Use off season for dumping F2. I would call it as broadcasting F2, something like your SSD. Just dump it, get one seed from it. So we are not, without losing time, I come to F3. I always consider F3 as the base for starting artificial selection. It's written in all books that you should start in F2. Why not in F3? You get these two big advantages. Extent of heterozygosity is reduced, and then the minimum population size reduces from 4 per locus to 3 per locus. Not even 3, it's less. We have done that calculation, which means the chance of getting the most potential transcriptional segregant increases substantially in F3. So
yeah, but now, the current trend which is happening in all the genomic selection models, right, is to go for development of recombinant dendritic lines, RDS lines, right? Like MAGE program, Prasanna was mentioning in the morning, using the double haploid facility, and using RGA, taking as many as 5 to 6 generations per year, and then fix your, say, 500 reels in one cross, and if you have 10 cross, you get 5,000 reels. So the point is that this is the approach being used where it is possible to capture the variation generated by recombination between two diverse parents. As far as major loci are concerned, you don't really miss. So that's where how is that possible there if it is happening?
Correct. Here, a lot of genotyping is involved, and that's why you think on these lines like, okay, 500 is all right. But according to the breeding principles, the required population size is large. Even though, so that's why I'm saying, if you are having to raise F2, F3, F4 in the field, you should not have difficulty in raising larger population in early generations. It doesn't cost you much as compared to genotyping. So we are not talking about a situation where genotyping is done. I'm talking about a simple breeder who is working in a small town with 2 lakh ORC. What he should do? I'm behind him.
I'm saying not 100.
Say something, Ashok, at this point of time. If you are producing double haploids, 500 of them, that makes your population size 50,000 F2 or sexually derived plants. And if you are handling number of crosses, then number of plants per cross and number of crosses, some counterbalance has to be worked out. Practicable and also economically feasible. You wanted the help for preparation for the cultural program. I think it is time that you take over and we conclude the session. And Professor Patel, I would like to thank you.
Thank you, sir.
On behalf of the audience, on behalf of the organizers, on my own behalf, profusely and convey that I really enjoyed immensely listening to you. I wish I could go on and listen, but cultural program has to occur. The hall has to be prepared, and also the participants have to have a few moments of walk around so that they are comfortable.
Right.
So thank you very much.
Thank you.
Thank you, sir. May I now request Dr. A.K. Singh to felicitate chairman, co-chairman, and to the speakers of the session.
is the camera?
Sir.
Oh. I will show it to Dr. B.D. Singh and then.
Okay. So,
Co-chairman, sir, if you are still there, so there is one mini bus for you, and I'm likely to come this month end to Varanasi. When I come, I will bring it with me.
Right.
Thank
But
you
I
very much.
accepted
Thanks for
today
being present.
virtually. I accepted. Thank you very much.
Thank you.
Thank you, sir. May I now request Dr. Rajbir Yadav for vote of thanks.
I think majority of you are late from the cultural program, and you are waiting that the session should be over. But it's informal. I will finish it quickly. Honorable Dr. B.D. Singh ji, our most valued invited speaker, researchers, students, ladies and gentlemen, it's my privilege to have been asked to propose a vote of thanks on this occasion. I, on behalf of Genetics Club, Indian Society of Genetics and Plant Breeding, Faculty of Genetics Division, entire agriculture researcher fraternity, and my own behalf, extend a very hearty thanks to Professor Balramji, such a respected and knowledgeable person, for agreeing to chair this important session. Your idea, sir, on the right of the other species beside human on Earth's resources is really thought-provoking. Sir, your words have inspired the faculty, seniors or juniors, participating researchers and students equally. I also express our sincere thanks to Dr. B.D. Singh ji, the co-chair, who graced us with his presence and guidance. Thank you, sir, for sharing us with your words of wisdom. I must mention our deep sense of gratitude to the excellent presentation and in-depth study on the dark genome, emergence of the functional genomic by Dr. P.K. Dal. Sir, your quote that dark genome is not in dark, but a gold mine is worth pursuing. Similarly, the presentation by Dr. C. Viswanathan on genome editing, on crop plants, and not only the presentation, but the practical pursuing that goal is praiseworthy. I also wish to express my gratitude to Dr. S.S. Patel for his extraordinary presentation on application of the population genetics, developing the plant breeding approaches. Your presentation has made this very difficult topic to understand for majority of the researchers very simple. And I hope that many of the young researchers at least will draw inspiration from your presentation and to pursue this dying field of the biometrician. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues of the Institute. We know their job was quite difficult, but they have executed well and have provided unflawed logistic support. Thanks to the audience for their patience in hearing. Thanks to all, and thanks all again.